Betul ya. Didn't you hear Amanda's moving back to Colorado? It's 2013 and the end of my life. Freshman year's about to plummet just a little harder, but it didn't cause we kissed on that Halloween night. I bleached half my hair when I saw Zoe on your vest, but it was orange from 3% peroxide thanks to you.
Yeah,
I
and I don't
needed
really want
to
to die.
cry,
Right.
I headed right to Kendra's. I hated you
And
and
probably bio.
that you knew
Who was that?
there's drama. Found a club for that where I met ya. Had a heart attack, yada yada. At the end, yeah, we burned, made a couple U-turns. You were it till you weren't. High school in Jakarta, sort of modern sport. Didn't you hear Amanda's moving back to Colorado? It's 2013 and the end of my life. Freshman year's about to plummet just a little harder, but it didn't cause we kissed on that Halloween night. I bleached half my hair when I saw Zoe on your vest, but it was orange from 3% peroxide thanks to you.
Yeah,
I
and I don't
needed
really want to
to
die.
cry,
Right.
I headed right to Kendra's. I hated you
And probably bio.
that you knew
Who was that?
there's drama. Found a club for that where I met ya. Had a heart attack, yada yada. At the end, yeah, we burned, made a couple U-turns. You were it till you weren't. High school in Jakarta, sort of modern Sparta. Had no chance against the teenage suburban armadas. We were a sonata, thanks to tight lip fathers. Yeah, living under that was hard, but I loved you harder. High school in Jakarta, an elaborate saga. I still hate you for making me wish I came out smarter. You don't hate your mother, so do I. Could've ended different than it did. We went to high school in Jakarta. Got a group assignment, I'll be your fast place. You text it out, but only call when you're with the face. I'm petty and say call me when you're not unstable. I lie and tell you I'll be getting drunk at Rachel's. I wasn't. She doesn't even drink, but I couldn't have you sit there and think that you're better. Cause you're older. Are you better now that we're older? High school in Jakarta, sort of modern Sparta. Had no chance against the teenage suburban armadas. We were a sonata, thanks to tight lip fathers. Yeah, living under that was hard, but I loved you.
I was told this is where you sign up for drama club.
Yeah, it sure is. Give me just one sec.
Comedy drama. I still hate you for making me wish I came
I am
out
more
smarter.
like
You love, hate your mother, so
Yes.
do I. Could've ended different then again. We went to high school in Jakarta.
Sorry, I'm Ben, by the way.
I'm Nicole.
I know.
Oh.
Natasha's moving to New York.
That's cool. I like that. Okay.
Probably sometime in August. And I'm spending the summer in Singapore. I'm so sad I can't tell you shit anymore. I made friends with Abby this year. We're moving in March or so. And although you bring me to tears, I'm glad that we gave it a go. High school in Jakarta, American summer. Had no chance against the Marxist girl with marijuana. I was your pinata, she was a star charter. Glad she gave it to you to heart, but I loved you harder. High school in Jakarta, I won't but I wanna. Ask
Same bed, but it feels just a little bit bigger now. Our song on the radio, but it don't sound the same. When our friends talk about you, all it does is just tear me down. 'Cause my heart breaks a little when I hear your name. It all just sounds like too young, too dumb to realize that I should have bought you flowers and held your hand. Should have gave you all my hours when I had the chance. Take you to every party, 'cause all you wanted to do was dance. Now my baby's dancing, but she's dancing with another man. My pride, my ego, my needs, and my selfish ways. Now I'll never get to clean up the mess I made. Oh, and that haunts me every time I close my eyes. It all just sounds like too young, too dumb to realize that I should have bought you flowers and held your hand. Should've gave you all my hours when I had the chance. Take you to every party, 'cause all you wanted to do was dance. Now my baby's dancing, but she's dancing with another man. Although it hurts, I'll be the first to say that I was wrong. Oh, I know I'm probably much too late to try and apologize for my mistakes, but I just want you to know I hope he buys you flowers. I hope he holds your hand, give you all his hours when he has the chance. Take you to every party, 'cause I remember how much you love to dance. Do all the things I should have done when I was your man. Do all the things I should have done when I was your man.
Oh no, did I get too close? Oh, did I almost see what's really on the inside?
Good morning, everyone. Are you guys ready for today's event? Don't forget to turn on your camera and use the virtual background provided. Go invite your friend to join us too because today's event will definitely be super fun and full of knowledge. Okay, well, I can see here that there's still a few participants. Let me ask something to you guys. One of them. Are you guys excited for today's event? Oh, and don't forget to have some breakfast. Uh, mm, I still can see a few participants here. Well, I'm going to say this again. Don't forget to invite your friends to join us too, because today's event will definitely be super fun and full of knowledge since there will be two speakers that incredibly super fun and super full of knowledge. You guys will get many knowledge from joining this. So yeah, don't forget to invite your friend. Okay, since I already see there's enough people here, so let me start the event right now. Good morning, Shalom, Om Swastiastu, Namo Buddhaya, goodwill and prosperous greetings to all of us. The Honorable Head of Faculty, Professor Dr. Kusworo Adi SSIMT, the Honorable Head of the Biotechnology Study Program Diponegoro University, Dr. Sri Pujianto SSI MSI, the Honorable Head of the Biotechnology Study Program Universiti Malaysia Sabah, Dr. Laki Goh Pohwah PhD, the Honorable of our Supervisor, Dita Putri Kumalasari SSI MSI, and also the judges for the selection of Aspire 2025. As well as all the finalists who we are proud of, thank you for your participation and giving your amazing ideas, creativity, and hard work into this competition. First of all, let us give our gratitude for the presence of God with an abundance of grace to all of us, so that we can gather at this event today. Finally, the moment we've all been waiting for has come. Welcome to the awarding ceremony of Aspire, Asian Sustainability Pioneers Competition 2025 in collaboration with Universiti Malaysia Sabah. Allow me to briefly remind you about the essay competition we host. Yesterday, we had 10 teams of students from universities in Indonesia to showcase and present their incredible works. Are you guys excited to find out who the winners are? Let's celebrate their incredible achievement together. Okay, before that, to boost our spirits, I have a tagline. You guys can follow me. If I say Aspire 2025, you can say Vision in Action, Insight for Impact. Okay? Do you guys get it or want me to repeat it again? Okay, I guess I will repeat it for once. Okay? Aspire 2025, Vision in Action, Insight for Impact. Okay? Let's start. In 3, 2, 1. Aspire 2025, Vision in action, insight, force, impact. Okay, thank you all for your enthusiastic response. It's truly inspiring to see so much positive energy from everyone here. I can see that the audience is already full of energy, so I guess let's begin. Okay, first of all, allow me introduce myself first. My name is Almas Nabilatus Salama as the Master of Ceremony of Aspire 2025 awarding ceremony. Before we start the event, allow me to read the series of events for today. First, there will be an opening. Second, welcoming speeches. Third, webinar by first speaker. Fourth, ice breaking. Fifth, webinar by second speaker. Sixth, lunch break. Seventh, awarding. And the last is closing. And also, I will deliver the guidelines at today's event. First, finalists must remain present until the awarding session concludes. Second, winner will be announced by the MC or a representative of the jury. Third, finalists who are announced as winners may have one representative unmute and deliver a brief message or impression for a maximum of 1 minute, of course, in a respectful of manner. Fourth, other participants are expected to show appreciation while maintaining order. And the last is, the winner will be contacted by the commit via email for verification and prize delivery. For the participants, don't forget to fill in the attendance form after the second speaker so you guys can get a certificate. Okay, let's move on. The first event is the opening by praying, according to our respective religions and beliefs. Let's begin. Prayer is never truly finished. Before we continue to the next session, the commit will take some documentation to capture these special moments for our records and memories. Okay, now first we can do a formal pose. Okay, first we're going to do a formal pose and then aspire pose like this. You guys can follow me if you guys can. Okay, for all the participants, so please get ready for the documentation session and please turn on your camera, of course. Okay, let's take, first we're going to do a formal pose. Okay, in 3, 2, 1. Okay, let's move on to the next pose. Let's do aspire pose like this. Okay, in 3, 2, 1. Okay, thank you everyone for your participation. Okay, next, we would like to invite the head of the organizing committee to deliver some speech. For Ms. Adeline Hartono, the time is yours.
Okay, thank you, Almas. So, dear the Honorable Head of the Faculty, Professor Dr. Kusworo Adi SSI MT, the Honorable Head of the Biotechnology Study Program Diponegoro University, Dr. Sri Pujianto SSI MSI, the Honorable Head of the Biotechnology Study Program University Malaysia Sabah, Dr. Lucky Goh Pohwa PhD, the Honorable RR Supervisor, Mrs. Dita Kumarasari, SSI MSI, the judges and the speaker of the Aspire 2025 webinar, respected organizing committee, and also all of the attendants, especially the remarkable participant of the ASEAN Sustainability Pioneers Competition or Aspire 2025. We are delighted to welcome you to the webinar that we have been built with the theme of Beyond Border and Beyond Limits, Collaborative Pathway for a Sustainable Future. This webinar is the final part of Aspire 2025 series, which is a space that we have created to all of us as a sustainable pioneers from all of nation. We firmly believe that to create a sustainable future, we cannot work alone. So hopefully we can share this forum as a golden opportunity to gain a new inspiration and expand your network to share knowledge, idea, build cross-border collaboration, and let's enjoy every session of this webinar. Also, ask a lot of questions that you are curious about to the speaker and transform the knowledge you gain today into a real-world action. So welcome to the Aspire Webinar 2025. We are very delighted to welcome all of you and hopefully you can be inspired to become an agent of change that can bring impact. Thank you.
Thank you to Miss Adeline for giving a speech. Please give a warm round of applause for her. Okay, we're now going to invite the head of the Biotechnology Study Program from Diponegoro University to deliver the next speech. For Mr. Dr. Sri Pujianto, SSI MSc, the time is yours.
Oke, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, the honorable speaker, Prof. Hermin Pancasakti from the Biotechnology Program, Diponegoro University, and Dr. Lakikoh Poh, Head of the Biotechnology Program, Universiti Malaysia Sabah, the honorable judges, Dr. Suhaini and Dr. Anto Budiarjo, committee member from the Biotechnology Student Association of Diponegoro University and University Malaysia Sabah, all Aspire ASEAN Sustainability Pioneer Competition participants. Today is truly a joyful occasion as we gather for the Aspire event. This morning, we will announce the winner of the competition, and before that, we will enjoy an insightful webinar featuring two speakers, one from the biotechnology program of University Malaysia Sabah and one from the biotechnology program of Diponegoro University. Last year, I had the opportunity to visit University Malaysia Sabah and meet Dr. Lakiko, the head of biotechnology program. During that visit, we made a commitment to collaborate not only among lecturers, but also among students, with the goal of learning to work and coordinate across countries, which has become a global necessity today. One of the outcome of that collaboration is today's Aspire event. The aim of Aspire to improve a platform for ASEAN students to present ideas, research, and innovation solution related to the SDGs, sustainability development goals, and to build intellectual network among students from various universities across Southeast Asia, and to encourage collaboration for a greener and more sustainability future. I would like to express my sincere appreciation to the organizing committee for their excellent work in making this event run smoothly. My gratitude also goes to the biotechnology program of Universiti Malaysia Sabah and the World Class University program, Faculty of Science Diponegoro University for their valuable support. May this event bring great benefit to all of us and continue in the years to come. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Okay, our sincere gratitude to Mr. Dr. Sri Pujanto SSE MSE for the inspiring speech and meaningful guidance. Once again, I'm going to say thank you to Mr. Puji, and please give a warm round of applause for him. Okay, guys, before we move on to the next session, let me ask you again one more time. Do you guys excited for being in here today, especially for the finalists? Let me search for the finalists first. Where is them? For the finalists, please turn on your camera and also use the virtual background that already provided. And also all for the participants, I would lovely to welcome you guys for this event today. And also, not only for the finalists, but also for the participant, don't forget to use the virtual background and turn on your background. And turn on your camera and use the virtual background. Okay. I'm still seeing no one from the finalists turning on their camera. So I guess I'm going to ask shoot one of them, okay? Let me see. Okay, for finalist number 4, Alia Haniatul Ifah. Can you turn on your camera, please? And using the virtual background it provided. Okay, or maybe, oh, I see someone. Alicia Suci Nurwiyanti, maybe you can unmute and I want to ask you a few things. Hello, Alicia.
Hello,
Hi. Do you have your breakfast today?
I do.
Wow. What did you eat today for the today's event? Do you excited?
Yeah, I'm so excited. I'm just eating some bread.
Okay. Let me ask you something. What do you prepare for today's event? I mean, you're a finalist, so do you prepare something special?
No, I'm not really prepared for the outcome for this. I'm really excited to know the winner, but I'm just waiting for the awardee later.
Well, I can hear that you say you cannot wait for the winner later, so I guess you need to stay in this teams until the announcement of the winner of in the awarding. So, okay, thank you for your answer, and yeah, let's see today's who's the winner. Okay, maybe after Alicia, I want to ask another finalist. Oh, I can see that Alia Haniatul Ifa already turning on her camera. Maybe Alia, can you unmute? Hi, Alia. Can you unmute, please? Well, I think Alia isn't there, so let's move on to the next person. Okay. Oh, I see. Finalist number 6, Arya Wijaya Adibrata. Can you unmute, please?
Hello.
Hi, good morning.
Morning.
Do you excited for today's event?
Yeah, absolutely.
Wow. Can I ask you, do you have something special to prepare for today's?
Uh, I would say not very much, but something for sure is that uh, I'm a bit nervous for who's the winner.
You don't need to be too nervous. Just pray for the best at the awarding session later, okay? But thank you for your answer. And yeah, stay tuned. Okay, well, I can see many of finalists and participants here who's already excited to knowing who's the winner is. So I think you guys must stay until the end to know who's the winner of this Aspire competition. Okay, I think without further ado, let's move on to the next session, which is the most interesting part. Okay, ladies and gentlemen, we will now move on to the next session. To guide us through it, it is my pleasure to introduce our moderator first. I will begin by sharing a brief overview of her impressive background. Okay, let's see. Wow, okay, as we can see from Amy's CV, she is currently in her second year studying biotechnology at Diponegoro University. Maybe we can scroll it down more. She is from Jakarta and she is also currently a member of the Student Senate organization at Faculty of Science and Mathematics Diponegoro University. Oh, maybe we can turn it down more, scrolling it down. Well, she has also participated in several volunteer programs such as Jentara Asa Community and being a volunteer at SDN Kalikesek and also volunteer in Nur Hidayah Orphanage. Wow. She has also been an MC at international seminar. Well, I bet that she's pro at it. Okay. So yeah, without further ado, please welcome Aimi Devian Najah.
Thank you so much, Almas, for the warm welcome.
Hi, Aimi. How are you doing today?
Hi, I'm very great to be here. I'm very excited. How are you?
Well, I'm pretty good too, obviously, since I'm here and I'm super excited to being in here. Okay, what have you been doing recently? What are you busy in?
I've been busy with a lot of university task assignments, so I've been just pretty much spending my time doing on them.
Wow, I heard before that second year is not an easy year, so yeah, I feel it too. Okay, how do you feel about moderating the event? Are you feeling ready and excited?
Yes, I'm very excited to be here. I think it will be a new experience for me, and it will definitely harness my skill in public speaking and in communication. So, yeah, I'm very, very excited to be here.
Wow. Okay, I can hear that you seems really ready to be moderator for today's. So yeah, let's just get straight into it. To Aimi, the time is yours. Okay, maybe before I'm going to moderate, there will be some videos to show. You guys can watch it and yeah, let's enjoy it.
Might turn to be main cause of every possible tragedy on Earth. Hence, a living creature should be observed for the sake of science.
Selamat datang di Fakultas Sains dan Matematika Universitas Diponegoro. Kami berkomitmen menjadi fakultas riset unggul berstandar internasional, mendukung Universitas Diponegoro bermartabat dan bermanfaat, serta mewujudkan World Class University ranking 500 dunia, dan mencetak lulusan berdaya saing global serta nilai moral dan etika yang kuat.
Accordingly, we put many scientists out, dedicated every single piece of our heart for each research. Assisted with sophisticated tools and advanced technology, no matter how complicated the situation is, it will be elaborated to bring out the solution. Compounds were studied, collected up to form a theory. Every single invention were integrated with another invention.
Melalui integrasi yang harmonis antara pendidikan, penelitian, pengabdian, dan kemahasiswaan, kami berkomitmen mencapai standar World Class University dengan keunggulan yang terukur dan berkelanjutan. Kami membangun ekosistem pembelajaran yang holistik, mengintegrasikan pengembangan akademik, keterampilan soft skill, dan hard skill. Penelitian dan pengabdian kami terpusat pada pengembangan solusi inovatif yang strategis dan berkontribusi nyata bagi masyarakat seperti mengentaskan kemiskinan, memperkuat ketahanan pangan, sekaligus mencetak generasi pemimpin yang visioner, berintegritas, dan berdaya saing global.
Giving wide possibilities for new circumstances to be explored, investigating the source and resources to find out novelty, and proceeding a sequence of practicums. Cultivating collaboration, domestically as well as abroad.
Ini adalah unit produksi produk inovasi yang dihasilkan oleh Universitas Diponegoro dari Fakultas Sains dan Matematika yang berbasis teknologi plasma. Ada dua produk yang sudah dilisensikan oleh Universitas Diponegoro ke PT Dipo Teknologi, Zeta Green dan Biozone.
Considering the wide range of science, therefore, having strong mutual relationship
with as many institutions as possible is the key for any kinds of successful research.
SEMEPSA, Research Center ini terbuka lebar bagi semua pihak ya, yang ingin mengembangkan riset-riset yang muaranya kepada pengembangan aplikasi biomonitoring maupun juga pengembangan sustainable aquaculture. Kita sangat membuka lebar untuk kerjasama.
Anything impossible turned to be possible. Change something insignificant into something worthy. What was only a plan transformed to be a considerable execution. Upon every lecture, whether in the classroom or in laboratory, students were driven in an embedded discussion.
Selama perkuliahan di sini aku ngerasa dapat banyak banget ilmu, dapat banyak banget pembelajaran, mulai dari pembelajaran di kelas bersama dosen dari sisi teorinya, tapi di sisi lain dari sisi praktikalnya aku juga dapat banyak banget ilmu melalui kegiatan praktikum yang menurut aku pribadi sangat baik sekali untuk menunjang aku di dunia kerja.
Ketika aku masuk ke Smart Building ini, pelayanan yang aku dapatkan sangat baik karena staf yang lain ini sangat ramah dan siap membantu apa yang kita butuhkan. Di samping itu juga fasilitasnya sangat memudahkan kita, seperti pintu yang dapat terbuka secara otomatis dan lift yang dilengkapi dengan sistem sensor. Terdapat juga ruangan yang cukup luas dan nyaman untuk mengikuti kuliah umum.
Supported with adequate facilities, the Faculty of Science and Math Diponegoro University adhere to inclusive education.
Di perpustakaan ini, aku mendapat berbagai macam fasilitas, yaitu ruang belajar yang nyaman. Selain itu juga, aku dapat dengan mudah mengakses media pembelajaran, seperti buku, jurnal, karya tulis ilmiah, yang dapat memperluas wawasan dan juga membantu aku dalam pengerjaan tugas aku.
Proved by providing enrollment for disabilities. Its students deserve the same opportunity to create their own bright future.
Dengan strategi peningkatan kualitas mahasiswa, optimalisasi fasilitas, dan pengembangan sumber daya manusia, kami menerapkan tata kelola efisien dan akuntabel, didukung teknologi informasi terintegrasi untuk memperkuat keunggulan institusi. Melalui kolaborasi internasional strategis dengan universitas terkemuka dunia, serta sektor industri, Fakultas Sains dan Matematika Universitas Diponegoro siap menjawab tantangan global, memberikan solusi berkelanjutan, dan berkontribusi pada masa depan Undip yang bermartabat dan bermanfaat.
We strongly believe that all the dream come true emerge from the soul that keeps vibrantly enthusiastic.
Well, I'm sure after watching the video earlier, we'll definitely get to know Faculty of Science and Mathematics better. Okay, without further ado, let's just get straight into it. To Aimi, the time is yours.
Once again, thank you so much, Almas, for the warm welcome. Allow me to introduce myself again. My name is Aimi Devaninaja, and I will be your moderator for today's event. It is truly a pleasure for me to moderate two extremely remarkable speakers who will share their knowledge on biotechnology driven solutions that support the achievement of the Sustainable Development Goals. I hope you are all as excited as I am for what we are about to learn and experience today. Before we start, I would like to deliver the SOP for today's event. The SOP, or Standard Operating Procedure, is a set of guidelines to help our event run smoothly and in an orderly way. The first one, participants must keep their microphones muted except during the Q&A session. Second, participants are expected to pay attention to the event proceedings until the end. Third, participants who wishes to ask a question must use the raise hand feature and may unmute their microphones only when permitted by the moderator. And lastly, participants are encouraged to actively participate during the Q&A session. Now that the SOP is clear, let's move to the highlights of today's event. For the Aspire 2025 webinar event, we are delighted to bring together two outstanding experts who will discuss how biotechnology plays a vital role in supporting the Sustainable Development Goals. As we know, biotechnology encompasses a wide range of disciplines, from agriculture and health to industry and the environment. Blue biotechnology, or marine biotechnology, is among the fastest growing areas, focusing on the potential of marine organisms to promote sustainability and reflecting the spirit of Sustainable Development Goal number 14, which is life below water. Without further ado, let me briefly introduce Professor Dr. Hermin Pancasakti Kusumaningrum, SSE, MSE, as our first speaker to start off today's webinar event. Professor Dr. Hermin Pancasakti Kusumaningrum, SSE, MSE, is a professor in the Biotechnology Study Program, Department of Biology, Faculty of Science and Mathematics, Diponegoro University. She earned her doctoral degree in microbial genetics from the postgraduate program at Gajah Mada University. Professor Hermin has taught a wide range of courses, including aquatic biotechnology, metagenomics, and environmental biotechnology. She has led the project Metagenomic Approach in detection of metallothionein genes in heavy metal-resistant bacteria for bioreporters and biomonitoring in polluted Semarang waters. Her notable publications include Bacterial-Based Bioremediation, a sustainable strategy for mitigating copper and lead contamination in aquatic ecosystem. Additionally, Professor Hermin holds several patents and intellectual property rights, such as a method for creating a metal distribution map of heavy metals and microorganisms in polluted waters for flood disaster mitigation. With such remarkable contributions to science and biotechnology, please welcome Professor Dr. Hermin Pancasakti Kusumaningrum to the spotlight. Okay. Good morning, Professor Hermin.
Good morning.
My name is Aimi, and I will be your moderator. How are you today, Professor?
Alhamdulillah, I'm fine, thank you. And how are you?
I feel great, thank you. Before we start, I would like to ask just a few questions. I see that your presentation is about marine biotechnology. Do you mind sharing with us what inspired you to choose this particular topic on marine genomics and biotechnology for today's presentation?
Okay, thank you so much. I choose it because first I love sea and the marine environment. So my dissertation was talking about microalgae also, especially with their genes that can be transformed into other organisms. And now on, I also have some research in the coastal development regarding with melagenomic, also the polluted water, and so on. That's all.
Okay. Wow, that is very interesting. Okay. Do you think that this topic is closely related to Indonesia? If yes, how so?
Yes, because almost 70% of Indonesia was consist of marine waters, yes, isn't it? And we have a lot of homework, yes, to um, to gaining the productive marine environment on Indonesia, and there's also a lot of problems that we have to face, and we have to searching for the solution uh, that I think we can do it together.
Wow, okay, that was very interesting. I agree that Indonesia is indeed closely related with this topic, since Indonesia itself is an archipelago with a massive biodiversity. Wow. Thank you, Professor, for the insightful. And now we are fortunate to hear from Professor Dr. Hermin Pancasakti Kusumaningrum, who will deliver her presentation on marine genomics and biodiversity biotechnological approach for ocean sustainability. To Professor Hermin, the spotlight is yours.
Thank you very much for the moderator. Do you hear my voice clearly?
Yes, Professor, we can hear you loud and clear.
Okay, thank you so much. The Honorable Dr. Lucky Kohua, Head of Biotechnology Program, Universiti Malaysia Sabah, as the guest speaker. And the Honorable Head of Biotechnology Study Program, Dr. Sri Pujianto from Diponegoro University, and also the Honorable Lecturer and the invitees and all of the students and participants who attend at this moment. Thank you very much. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. I'm Hermin.
Warahmatullahi wabarakatuh.
Thank you so much for having me here today. I'm really excited to talk with you about marine genomics and biodiversity, biotechnological approach for ocean sustainability. And what is that? Yes, let's find out. Okay. Please go to the slide 2, please. Okay. This is the introduction, yes, that ocean covers 71% of the Earth's surface and home to vast biodiversity with unique genetic resources. And also marine genomics helps us unlock molecular secrets of marine life. And biotechnology, of course, offers tools for conservation and sustainable use. And slide 3, we will talk about how this marine genomics relating with Indonesia. We can see from the figure that I gave you all the importance of geographic position of Indonesia, that Indonesia is the only country in the world that is located along the equator between two continents and connect two oceans. Okay, that is very specific, yes, and was have a lot of advantages for us. Okay, we will go to the next slide. And we can see from the strategic position that Indonesia has very high biodiversity, where Indonesia is the largest archipelagic country in the world. You can see how much species that we have, yes, and a lot of island, also land area and water area. Indonesia is also known as the center of the world coral triangles, and Indonesian ocean hosts exceptionally high marine biodiversity, including coral reef species and fish and marine mammals. And Indonesia is home to over 3,000 fish species and 600 coral reef species. making it one of the richest marine ecosystem in the world. Okay, we go to the next slide. So we will go to the main topic with me. What is the marine genomics? Yes. Marine genomics was study of marine organism at the genome level. Genomics can be defined as a discipline in genetics concerned with the study of genomes of organisms and the outburst of chromosome, gene, and DNA. Genomes, on the other hand, are the entirety of an organism's hereditary information, which is either encoded in the DNA for many types of virus, also ribonucleic acid, and include both of the genes and the non-coding sequence of DNA or RNA. It can be seen as a branch of molecular biology that related with the study of the structure, function, expression, evolution, and mapping of genome of organism. The field includes intensive efforts to determine the entire DNA sequence of organism and fine-scale genetic mapping efforts. It is always generates large data set from cytogenetic, molecular genetic, quantitative genetic, and population genetic, and has led to the development of bioinformatics, through which raw genome information links to meaningful biological information. And the tools that was used to study marine genomic is DNA sequencing, transcriptomic, proteomic, metagenomic, and so on. Marine genomic is an emerging field of biotechnology that uses genomic technique to study and manipulate organisms in the ocean. It is very powerful tools for the conservation and management of marine environment. And also marine genomic generate insight for the production of novel products such as nutraceutical and marine-based biopharmaceuticals. And marine genomics also play an important role in climate change research, the impact of change to the ocean of organism and ecosystem. And marine genomics also improve the management and conservation of marine environment, sustainable use and preservation ecosystem. So it is needed to improve the management and conservation of marine environment using a lot of technology to sustain our marine genomic sustainability. Okay, and then we go to the slide 6. We can see that marine biodiversity have a lot of coral reefs, mangrove, seagrass, fish, deep sea ecosystem. And also marine biodiversity have rich source of novel genes and enzymes. The recent studies have uncovered new gene cluster and enzyme from marine microbes, and it includes 92,450 protein coding sequences. In 2020 alone, more than 1,500 new compounds were isolated from marine species. While nearly half of the approved pharmaceuticals are based on biological compounds produced by living organisms, success rates of 2 to 4 times higher for compound from marine organisms. And some factors that pose challenges and threat to a sustainable marine economy include global climate change, overfishing, marine waste, and debris pollution. And specifically in Indonesia, also unsustainable coastal development, marine habitat loss, illegal, unreported, and unregulated fishing, and also lack of infrastructure and technology. Okay, we will go to slide 7. We will discuss in more detail what is the threat to ocean biodiversity, and Indonesia also face the same problems. That climate change, such as rising sea temperatures, will increase acidity levels and change in rainfall patterns, which undoubtedly impact Indonesia marine economy. Rising sea surface temperature in Indonesian water can cause fish to migrate to cooler water. This can reduce fishermen catches in certain area and disrupt the sustainability of local fisheries. Furthermore, increased ocean acidity caused by rising carbon dioxide level can damage coral reef in Indonesia. Climate change can also lead to increased storm intensity and frequency, potentially will damaging marine industrial facilities, fishing vessel, and marine tourism infrastructure. We have seen a lot of problem in Pantura, yes, right now regarding with the flood and also how seawater came into our city, yes, and rising sea levels caused by climate change can lead to flooding in coastal area of Indonesia. Another impact of climate change is the shrinking of mangrove ecosystem, resulting in the loss of vital habitat for fish, birds, and various other animals. Furthermore, climate change can affect marine environmental conditions, including ocean currents and circulation patterns. This can increase the risk of marine pollution due to the spread of waste and hazard material that can damage ecosystem and threaten human health. Overfishing occurs when the rate of fishing exceeds the natural reproductive rate of a fish population, threatening the survival of fish species and disrupting marine ecosystem. And marine pollution is caused by industrial waste, agricultural waste, and plastic waste. The amount of plastic waste from Indonesia dumped into the ocean reaches 56,333 metric tons annually each year. And the 2021 Village Potential Survey in Indonesia shows that a total of 1,511 villages directly bordering the sea throughout Indonesia still dump waste into rivers. So they dump waste into rivers, irrigation, channel, lakes, and the sea. And also excessive use of pesticide and agricultural chemical can pollute water and damage biodiversity in Indonesia. If coral reef, mangrove, and mangrove or destructive fishing practices are damaged, Indonesia is estimated to lose 2,000 small islands, which could potentially shrink by 2030. Okay. And we will go to the next slide. Okay, we will see how biotechnological approach can overcome our problem, yes, that we have to face in Indonesia by using metagenomic, bioremediation, marine nanobiotechnology, and also bioinformatics and synthetic biology. As you can see of the definition on the slide, for metagenomic, that was study microbial communities in seawater or sediments, it is now widely accepted that less than 1% of microbes can be isolated using traditional culturing techniques. So 99% of microbes only can gain by using metagenomics. And this is leaving the vast majority of microorganisms and their biochemical pathway inaccessible. So this is the good choice for us to move right now into the metagenomic technology or environmental DNA. The emergence of culture-independent and metagenomic technique, however, provide us with the tool to determine the full extent of the uncultured microbial diversity and allow access to the biochemical pathway within this uncultured microorganism. There is a lot of microorganisms that can be uncultured in our laboratory. To date, 30,000 samples from 200 base stations containing millions of organisms have been collected from the ASEAN project. As a result, novel sequences from viruses, prokaryote, and picoeukaryote have been discovered. Start from 2010. We also can see the example of metagenomic research on slide 9. Next slide, please. Yes, this is the example of some of our research regarding to the metagenomic that you can see so a lot of species that we can get when we using metagenomic research. And when we comparing it with the culturing microbes, we only have maybe 3 until 10 isolate. But from this, with this technique, with metagenomic, we have almost thousand of microbes, yes. And we get it, this bacteria that was growth on the biofilm and the concrete and wood surfaces. Next slide, please. Okay. We can see from this figure that there's a lot of genus that involve in to form biofilm, yes, and several surfaces in the marine environment. And we found that the dominant genus in wood is Vibrio and Pleurocapsa. And the abundant genus in the concrete is Vibrio and Ruegeria. So we can conclude that Vibrio can grow dominantly in both surfaces. Okay, next slide, please. And from metagenomic analysis also, we can predict what kind of the pathway or the metabolic pathway that the microorganism have. And what is the pathway that relating to the ability to produce biofilm? And this is the result of that. Okay. Next, please. Okay. We move to bioremediation. That was eco-friendly solution, yes, for waste and pollution management, especially bioremediation using microbes. For example, heavy metal are widespread pollutant that can exert a selection pressure on microbial population due to their toxicity and persistence, leading to the evolution of heavy metal resistant genes. These genes are part of the resistome, and their spread often occurs via mobile genetic element that allow co-selection with antibiotic and biocide resistant genes. From the figure that on the slide, we can see that resistant gene to some heavy metal is specific to gram-negative or gram-positive bacteria. And the genes that regulate it are located in the operon system. Arsenic, chromium, lead, copper, and mercury are associated with both gram-negative and gram-positive microorganisms, respectively. The specific resistance system for gram-negative bacteria was with zinc, cobalt, and cadmium. And for gram-positive bacteria, they related with copper and cadmium. In the eukaryotic system, for example, microalgae includes fungal resistant to arsenic, chromium, lead, cadmium, and copper. Next slide, please. Okay, this is the example of marine nanobiotechnology that we had conducted several years ago. And we choose this because pollution in marine water is a long-standing environmental problems that poses a risk to both the environment and human health. And we choose to searching for a solution by using the marine nanobiotechnology. Okay, on the next slide, please. This is the nanoparticle of microalgae that they absorb the heavy metal. So they reducing the heavy metal in the marine environment. And also freshwater and marine water also, as you can see in the picture, that microalgae that you use was able to accumulate metals and reduce metal ions so that they are widely used for nanoparticle biosynthesis in living and dead condition. Next slide, please. Okay, this is application of metallic nanoparticle in aquatic environment. You can see that some cell will be experience lysis, but there are also those some microorganisms that are able to survive by carrying this nanoparticle of heavy metal in their cell, depending on the nanoparticle that they enter to the cells. Actually, we want to know in this research what kind of the heavy metal particle that affect the cell of this microorganism. Okay, next slide, please. Okay, so we will discuss about bioinformatics. Bioinformatics was the solving problems that arising from biology using methodology from computer science. They have the concept from genomic, proteomic, pharmacogenomics, and microarray. Okay, as you can see that multi-level approach based on the molecular investigation are essential to discover marine resources and further explore key molecular processes involved in their production and action. As a consequence, omic approach accompanied with the associated bioinformatic resources and computational tools for molecular analysis and modeling are boosting the rapid advancement of biotechnologies. Next slide, please. Okay, this is some case study for marine microbes producing plastic degrading enzyme. Maybe I give an example from metagenomic data that some researchers have found some notable marine microbes that can degrade plastic, for example, in the marine environment. For example, Escatobacter, Rhodococcus, Streptomyces, Vibrio, Olispora, and others. Okay, and then the algae engineered for biofuel production. The famous microalgae is Chlamydomonas reinhardtii. It was engineering to produce the biofuel, and there's a lot of microalgae also instead of the Chlamydomonas reinhardtii. And for coral genomic research to improve resilience to climate change, that the intensive study of coral bleaching in Indonesia, especially on Undip, yes, we can see it as the clearest threat to climate change, to the persistence of marine biodiversity. But some coral may be able to rapidly adapt to warming ocean temperature. For example, is Acropora hyacinthus. This coral possesses 78 identical genes that respond to heat stress, that some were upregulated and others were downregulated. Okay, next slide, please. This is the microalgae that was identified using the TUFA gene. Why we use this TUFA gene? Because chlorophyta, yes, is very difficult, yes. When we because chlorophyta is represent a major biodiversity component of eukaryotic microalgae, and identification and classification of this group through morphology is very hard, since it might present cryptic species and phenotypic plasticity. So when we do not identify by using the molecular barcoding, we will get the wrong identification, wrong name, and it makes us difficult to improve their potency. So according to a lot of researchers, that the TUFA marker stand out as a good choice to be used as molecular marker for chlorophyceae. Okay, so I have experience when I want to buy the Hematococcus pluvialis from several known places in Indonesia. We do the identification using the molecular marker. Almost all of them have wrong identification, and I didn't get the Hematococcus pluvialis, but instead of Chlorella surukiniana. Just like that, because the plasticity of phenotypic of the chlorophyceae. Next slide, please. Okay, this is the use of the macro and microalgae for biofuel generation. We have one generation, second, third, and the fourth generation. And I will explain only third and fourth because of the time. I think that let me see the third generation using aquatic feedstocks such as algae biomass. But however, it is have disadvantages such as high cost and the fact that biofuel produced from algae is less stable and then that produced from other sources. And we came into the fourth generation biofuels, which are still in early development, use bioengineering microorganisms such as bioengineering algae or crops that are genetically altered to consume more carbon dioxide from the environment. And they will use to produce the ethanol, butanol, hydrogens, methane, vegetable oil, biodiesel, isoprene, and others. Next slide. Okay, we have oceanomics, yes. It's very recent development, yes, that a primary goal of this oceanomic is to develop environmental DNA as a cost-effective, scalable biomonitoring tools. And the key activities include producing environmental DNA biodiversity and high-quality reference genome dataset at scale, making this data available and translating information for communities and to influence policy and decision-making framework. Okay. And next slide, please. Okay. We have the Ocean Sustainability Goals. Yes. United Nations for Sustainable Development Goals 14 that states life below water. We can see that because of human action have altered 66% of the ocean and the triple planetary crisis of climate change, nature and biodiversity loss, and pollution and waste is exacerbating the impacts. Okay, and how to protect a marine ecosystem using genomic insight? We will do it by analyzing ADNA that found in seawater samples. We can use it to track endangered species, monitoring ecosystem health, and detect invasive organisms before they threaten native population. These breakthrough techniques provide real-time insight into ocean health while acquiring minimal disruption of marine life. And how to promote the sustainable blue economy? Blue biotechnology provide opportunities for a wide range of initiative of commercial interest for the pharmaceutical, biomedical, cosmetic, nutraceutical, food, feed, agricultural, and related industries. Next slide, please. Okay, we will go to the challenge and future perspective. We will talk about ethical concern and bioprospecting regulation. Indonesia have Nagoya Protocol regarding with this. And also we have Undang-Undang Republik Indonesia number 1, 2014 about pengelolaan wilayah pesisir dan pulau-pulau kecil. And we also have the Peraturan Pemerintah Republik Indonesia number 82 about pengelolaan kualitas air dan pengendalian pencemaran air. And what is the need for global collaboration and data sharing? Of course, it is recommended to establish a joint consultation mechanism composed of scientific researcher, research funder, platform builders, data users, and representative of the public through the relationship network. Okay, maybe we need to form the Marine Information Center and Ethic Review Committee. Okay, and then and how to use the artificial intelligence and bioinformatics to accelerate marine genomics. We can use DNA barcoding and eDNA to monitor marine biodiversity. And also We can use it to identify and track population change by collecting and analyzing genetic material from water samples, making it possible to detect marine life without direct observation. And biotechnology as a key driver for future ocean sustainability. As stated in the previous slide, marine biotechnology is recognized as a globally significant economic growth sector. The last slide, please. Okay, we have conclusion that marine genomics is essential for understanding biodiversity, and biotechnology provides powerful tools for sustainability in protecting ocean, ensures the health of our planet, especially for Indonesia. And preserving the genome of the ocean means preserving life on Earth. Allow me to conclude the presentation by quoting Darwin's statement. It is not the strongest nor the most intelligent, but the species most adaptable to change has the best chance of survival. Thank you for the opportunities. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, everyone, please give a round of applause for Professor Hermin. Okay. Wow, that was truly an insightful presentation from Professor Hermin. It is interesting to see how the collaborations of different fields of biotechnology contributes in sustaining and conserving our oceans. Before we proceed to the Q&A session, allow me to share a brief summary of the main discussions and key takeaways from this presentation. This presentation highlights the critical role of marine genomics in understanding and conserving ocean biodiversity. Marine genomics is the study of marine organisms at the genome level and is vital because of the presence of threats to ocean biodiversity, such as pollution and overexploitation of marine resources. Biotechnology's approach to these threats are through metagenomics, bioremediation, marine nanobiotechnology, and so a lot others. The metagenomics approach is through taxonomic composition comparison on biofilm. Bioremediation utilizes microorganisms to solve pollution problems, and marine nanobiotechnology develops nanoparticles for remediation. Some case studies are also presented showcasing the application of biotechnological approaches to real-life cases and how it supported the SDGs number 14. It also highlights how biotechnology provides powerful tools for sustainability. Now, we move to the Q&A session. For any participants who would like to ask, please use the raise your hand feature, and you may use the microphone, or if that's not possible, please type your question in the chat room. All participants are encouraged to be active in this Q&A session. Whilst we have the expert here, why don't we go for it and ask the questions that you want to ask? It's a very interesting presentation, especially on how it fits really closely to us being here in Indonesia. We have our own massive biodiversity and everything.
Or maybe we can go to the next speaker first, and we will get a question after the speaker finish. The second speaker finish.
For this, we will actually just hold the Q&A session first, and then the next speaker, Professor.
Okay.
So anyone who wants to ask a question, please don't hesitate to raise your hand or through chat.
Uh,
Okay, for Nurlita Anggreni, you can unmute your mic now and ask away.
okay. Good morning, Professor Helmi Pankasakti.
Good morning.
Thank you for the insightful presentation. My name is Nurita Anggraini from the Biotechnology Program at Diponegoro University. I would like us to about before that, I'm very interested in biofuel topic using microalgae, considering that global warming significantly affects marine ecosystem by increasing ocean temperatures, which should be prioritized, Prof. Exploiting marine resources for biofuel production or restoring marine ecosystem to mitigate the impacts of climate change. That's a question for me, Prof.
Oh, okay. I'm sorry. Can you repeat it once more?
Should I repeat my question?
You asked about the relationship between biofuel from microalgae to climate change, right or not? Or maybe you can ask in using bahasa. It's okay also. Can moderator help me for the question?
Okay, Nurlita asked the question, um, which one should be prioritized, exploiting marine resources for biofuel production or restoring marine ecosystems to mitigate the impacts of climate change? Is that clear enough, Professor?
Yes. Okay. I'm sorry to misunderstanding the question. Yes. In my opinion, yes, the most important things because right now we still have a lot of microalgae, a lot of algae to produce the biofuel. So we can choose the second one. While using our microalgae, we also can wait. Because Indonesia is facing the climate change a lot due to the tropical country. Yes, so we have to choose this first to overcome. And this is also the benefit, yes, because we have the tropical climate, so the microalgae or the algae will reproduce a lot, yes, to use as biofuel. Is this answer your question?
Yes, Prof.
So-
Yes, enough. Thank you, Prof.
My pleasure.
Okay, we have another question from the chat from Salman Jafar. How do techniques like DNA sequencing, transcriptomics, and metagenomics enhance our understanding of marine biodiversity?
Okay, thank you so much for Salman question. Because Indonesia was or having a lot of biodiversity, yes, mega biodiversity, and we do not know right now, yes, about the data, a lot of data from our mega biodiversity, because 99% of them for microbes cannot cultivate it, yes, especially from marine microorganism. So the technique sequencing, transcriptomic, metagenomic, and so on was needed, yes, needed to develop the data from Indonesian biodiversity, because also it was cost effective and also high scalable biomonitoring tools, and also it was the key activities on producing a genome dataset for high quality reference for us. And we want to making this data available, especially from Indonesia resource. Yes, and also we want to translating this information to influence the policy and decision-making frameworks to our government. Okay? Is it answering the question from Salman?
Salman, I hope that answered your questions. Okay, thank you for the questions. Now we have the third and last question. How from Aushaf Zaki Hakimul Bahri, how can biotechnology approaches help maintain the sustainability of marine ecosystems?
Okay, thank you so much for a question from Zaki. As I have explained before, that biotechnology was consists of a lot of methods and a lot of new technology, especially metagenomics, yes, that will help us to having a lot of data, yes, that this data very potential, that can be used for conservation, for improvement, and also for searching a lot of new genes and enzymes to produce the new nutraceuticals, and also for human health. And I think this is a lot of advantages will make us having the sustained marine ecosystem, and it will have the biotechnology method like and in recent days will help us a lot. Okay.
Okay, to Aushaf, I hope the question is answered nicely. Well, okay, because the time for the question and answer session has run out, we thank for all participants who has volunteered to be active and as active during the Q&A time. Before we officially close this session, we would like to present an appreciation to Professor Dr. Hermin Pancasakti Kusumaningrum for her contribution to today's webinar session, followed by a quick documentation moment. We invite Ms. Dita to come and present the certificate of appreciation to our distinguished speaker.
Okay, so we would like to express our gratitude to our speaker, Prof. Hermin. Thank you so much. for your time and for sharing such a valuable insight and knowledge with us today, Prof. Thank you so much, Prof. Hermin.
My pleasure, Budita.
Okay, so it's very interesting knowledge and valuable insight for us to know about the marine biotechnology, especially as your expertise. And thank you so much, Prof. Hermin, to join with us today.
Okay, you're welcome. Thank you for the opportunity to ask me
Okay.
to join this venue.
Okay, thank you so much, Prof.
Okay.
Okay. Before we officially close this session, we would like to do a little quick documentation moment. For participants on Zoom, if it's possible, you may turn on your camera. We will have a little documentation moment with our distinguished speaker, Professor Hermian, to the documentation division. I will count from 3, 2, 1. 3, 2, 1. 3, 2, 1. Okay. Thank you so much. On behalf of the committee, I would like to express our sincere gratitude to Professor Hermin for the insightful and inspiring presentation. To Professor Hermin, if you have any other agenda after this, you may leave the Zoom room kindly. Thank you very much, Professor Hermin.
Thank you very much.
I'm sorry, Professor Min, for this digital certificate, the real one with the signage from the deacon will be sent later. Thank you.
It's okay. I'm sorry, I have to leave now. Thank you very much for all.
Yes, it's okay. Thank you, Professor Hermian. Okay, that was a really fun first session. I hope that everyone was hyped up already because we have another speaker, also an expert in his own skill, in his own expertise, and I cannot wait to introduce this second speaker. I hope that everyone is still excited and is still here in the Zoom room. And I hope you guys stay excited. Grab your food, your drinks, and your notebooks in case you want to write down the notes and everything. Okay. Let's return to our main agenda. We are excited to continue with our second speaker, who will bring another valuable perspective to fuel today's discussion. Earlier, we had the opportunity to explore blue biotechnology. In this session, we will discuss red biotechnology or health biotechnology. While blue biotechnology emphasizes the utilization of marine resources for sustainability, red biotechnology focuses on medical research, healthcare innovation, and the advancement of human well-being, aligned with Sustainable Development Goals number 3, which is good health and well-being. Please allow me to introduce Dr. Laki Gufahua, PhD, as our second speaker. Dr. Lucky Gopahwa is the head of the Biotechnology program and a senior lecturer at the Faculty of Science and Technology, Universiti Malaysia Sabah. He obtained his PhD in biotechnology from Universiti Malaysia Sabah. Dr. Lucky's research expertise spans drug discovery from microorganisms as well as epidemiology and genetic polymorphism studies, employing molecular biology techniques such as PCR, RFLT, and Sanger sequencing. He is also an active member of the Good Practice in Traditional Chinese Medicine Research Association. Dr. Lucky is currently working on the project Quantum Dot-Based FRET assay for the detection of virulence gene in Acinetobacter baumannii. In recognition of his contributions, he received an award at the International Congress of Blood and Marrow Transplantation in 2020. He also has published widely with notable works including detection of coronavirus in viral transport media using ultraviolet and near-infrared absorbance spectra and pattern recognition model. With his extensive expertise in biotechnology, please welcome Dr. Lucky Gopahwa to the spotlight. Okay. Good morning, Dr. Lucky.
Yeah, morning, moderator.
Okay. My name is Aimi, and I will be your moderator for this session. How are you today?
Yeah, things great.
Wow. I'm actually very excited about learning new things. I see your presentation is about healthcare and its role in improving lives. If you don't mind me asking, what do you consider to be the most urgent issue that inspired you to bring this topic today?
Is I think nowadays people are more and more aware of health, especially in terms of communicable and non-communicable diseases. So that's why this topic actually will more relate to us, to our surrounding, and to ourselves.
Okay. Do you think that biotechnology will continue to be one of the leading sectors in solving healthcare issues?
Yes, so talking about healthcare and in the context of biotechnology, so the main or the fundamental definition of biotechnology is to use technology to improve the life and the well-being of the humans. So by utilizing all these technologies that we have, especially what Prof. Nirma has presented just now, so that is actually part of it where it addresses the climate change as the main. So in the whole, it will improve our healthcare as well.
Wow, okay. Do you think that someday biotechnology will collaborate with other sectors to improving healthcare for sustainability as well as in achieving the SDGs number 14?
Yes, so when we talk about healthcare, it is not only biotech research. So it's more towards how clinicians, the doctors in hospitals works together with the researchers and the academia. So to later, I think in my presentation, so I will share how we work with clinicians. Of course, the main research is in biotech, but we work with clinicians and the doctors.
Wow, okay. Um, in in Malaysia itself, how is the role of biotechnology plays in healthcare? Has it been implemented in Malaysian healthcare?
So in Malaysia, we recently have a new National Biotechnology Policy 2.0. So that actually comprises a lot of aspects, including healthcare. So it looks into in terms of the policy, in terms of the funding, and in terms of the research focus that is currently required.
Okay. Well, thank you, Dr. Lucky, for the preface perspective. Now, to dive deep into the topic further, we invite Dr. Lucky to share his presentation on healthcare biotechnology and the SDGs, innovating for better lives. To Dr. Lucky, the spotlight is yours.
Right. So, first of all, good morning to everyone, especially the moderator Amy, Honorable Prof. Hermin, and then the Undip team for inviting me to give this talk. So, the talk that I will be delivering today is mainly focusing on one of the SDGs as well as healthcare biotechnology. So throughout it, I will give some examples of how when I'm doing biotech research, so it also addresses the SDG. Next, please. So this is where I came from. Okay, so from North Borneo. Okay, from Sabah. So University Malaysia Sabah actually has 3 campuses. So one is in the Labuan, which is a financial campus. Another one is in the Sandakan, which is the sustainable agriculture campus. And the one that you see in the middle is the main campus, which is in Kota Kinabalu. So I think some of the team from Undip from the program came here. So then we had a meeting and we discussed a few things. So it in the main campus, so we are spanned by in the front, so we can see all the seas, and at the back of the university, so we can see the mountains. So that's why we have 2 different views in the morning and in the afternoon. Yeah, next, please. So these are the people behind the scenes, okay, which led to where am I and who am I today. So in terms of giving this talk, so the team actually from UMS and the people that I've worked with together, especially the clinicians and my postgraduate students, my research assistants, and my colleagues. So we work together in terms of how we want to address the healthcare biotechnology, that's the main, as well as looking at biotechnology as a whole in Sabah itself. Next. So how does biotechnology and healthcare relate, right? So the most direct relation is to disease detection. So when we talk about diseases, so first it's either we prevent, we detect, we treat, or we mitigate. So how we prevent, okay, is through biotechnology studies, okay. So and then how we detect. So in the detection itself, so most of the biotech tools is focusing on molecular diagnostics. So whether it is using PCR for gene detection, microarray, or ELISA. So all this detection will lead to early disease detection and improve patient outcome. For example, in the context of cancer itself, so cancer, it needs to reach a certain size, let's say for colorectal or breast cancer, right? Then only the hospitals, the doctors, when they do MRI or when they do CT scan, so they can look at the images. So that will be followed by histopathology, where they take the biopsy samples. So that is a standard healthcare practice. Okay, so when it works with biotechnology itself, and in this slide particularly is looking into the molecular diagnostics. So what genes are there? Let's say the most famous one that we know, the well-known, is the BRCA gene for breast cancer. So the BRCA1 and BRCA2. So that is for if the certain individuals have a certain point mutation or certain single nucleotide polymorphisms, SNPs, then it is that particular individual have a higher risk towards breast cancer. That is for BRCA and breast cancer. So in colorectal cancer, there are a few also. So depending on the cancer type, okay, so early screening can be done through molecular diagnostics to know what are the markers and what are the risk factors. Okay, so in my next slide, okay, where how we solve global health challenges, right? So on top of non-communicable disease, which are cancer, diabetic, high blood pressure, those non-communicable. So we have another group of diseases which are communicable diseases. So those the most recent one, of course, that we know is COVID, right? So that are communicable diseases. Of course, then throughout the time, then we have a different influenza strain, we have a different vaccines that is required, right? So in infectious diseases or communicable diseases, okay, so a lot of times it's human to human transmission or human to animal, animal to human. So those are the zoonotic transmissions, right? So where we see that there are different cycles of zoonotic transmissions. So that actually leads to infectious diseases. And currently there are a lot of outspill. So means that if the forest has been undergo deforestation, so the disease would outspill, overspill, right? So means that the disease actually is contained in that environment because of deforestation and because of urban development. So the animal carrying the diseases is closer towards the human, right? So then that will lead to human and animal interaction, which leads to the outbreak of several diseases. So then that would actually contribute to antimicrobial resistance because antimicrobial resistance is one of the hot topics, okay, recently. So because yes, we have antibiotics to treat bacterial infections. We have virus drugs to treat viral infections, right? But those drugs actually, because of misuse of drugs, misuse of antibiotics, so then so the bacteria or the viruses are getting more and more resistance. So in my next slide, so I will present something that what our team has done, okay, especially me and one of my postgraduate students. So we are using quantum dots, okay, to detect a certain DNA marker. Okay, so in the next slide, okay, so what we see is that currently we know that molecular diagnostics is good, is the gold standard. But considering the time, we need to do nucleic acid isolation, and then we need to do PCR, and we need to visualize the result, and we need to QC. That actually would take you probably around 2 hours to 3 hours. So what we have presented here in this particular slide is we use quantum dots, okay? Quantum dots actually is a fluorescence compound, okay? That fluorescence, okay? So we conjugated the quantum dots and the graphene oxide, okay, with nucleic acids. Okay, depending on what are our target. So it can be a certain resistance gene we want to detect, or it can be a certain protein that we want to detect. Okay, that we can use aptamer, okay, to conjugate it on the sensor itself. So then we characterize using FTIR, TEM. Okay, so in the next slide, okay, I'm showing you is the characterization of the slide of the quantum dots and GO. So on the left side is the FTIR to show that there is changes in the absorbance, showing that the amine bond has been formed for graphene oxide, which is GO, and quantum dots QD. Okay, so on the right side is we use transmission electron microscope, TEM. Okay, it's a powerful microscope to look into specifically, are there any morphological changes after we conjugate compared to before to confirm that our conjugation of the capture A and capture B is successful. So we have shown that in these results, there are changes in the FTIR as well as in the TEM results. So we can see that it's successfully conjugated. So then after successfully conjugated, next is we looking into what are the highest quenching efficiency and limit of detection in my next slide. So we have successfully looked into the highest quenching efficiency, which is 58.5%, okay, and the limit of detection is 0.2. So what we are detecting here is the DNA molecule, okay? So of course we have to isolate the nucleic acids, okay, and then with the nucleic acids, we mix with our quantum dots and graphene oxide. Then we check under the fluorescence spectrophotometer, okay, to get all these absorbance and reading. And then we reconfirm back with our PCR products, okay? So that's why we were able to distinguish between complementary and non-complementary sequences. So with a statistical significance, meaning that our assay is specific, okay, towards it. And we showed that when we use clinical samples from the hospital as well as synthetic oligos, so it shows similar quenching efficiency. So it means that in this context, in my next slide, so we have actually developed a method, okay, which then we will look into it, how we want to develop it into a biosensor base. It's a point of care testing device, okay, which is we are currently working on developing one device, which is particularly for this, so to develop a biosensor. So in my next slide, and next, okay, so we look into the diagnostics, right? So it's a lot, right? So we have personalized medicine, and then we have artificial intelligence in bioinformatics for drug discovery. So that is more towards when we look into AlphaFold by which is a software that can predict the folding, tertiary folding of the protein, right? So then with that tertiary folding, then we can do molecular docking and dynamic simulation to see what chemical compounds actually can bind to the drug, right? So in order for a drug to work, in order for antibiotic to work, so it needs to bind. Okay, so that's why, in fact, we have done that. So, but because of time, I'm not presenting that. Okay, so we have done that. We have molecular docking. In fact, is using algae chemometrics data. Okay, so we take the list of compounds that can be isolated from algae, and then we have a particular protein, okay, which is looking into immunotherapy. So that protein actually PLD, okay, so is actually responsible for immunotherapy, okay. So then we look into has a few target candidates of the chemical compound, and we have found that. So that actually save a lot of time, okay, comparing to conventional natural products, screening isolation method, where you need to have thousands of pure or crude isolates or crude compounds. Then you test on the isolates itself, then to see which one is the most potential. So now what we do is we use computational method, okay, which is very fast. Okay, we can do it in 1-2 weeks time, given that everything is ready. Then we will have the result of what compound actually is significant. Then we just test that few compound instead of tens of thousands of natural products that we want to test one by one. But now we screen using computational method is much faster. So then in the next slide is during COVID-19. Okay, so we have very good in sequencing. Okay, looking at the variants of a lot of variants, okay? So that different variants comes from next-gen sequencing using Illumina platform, right? So of course, that also leads to development of several rapid detection method, okay? Like the one that we use is a rapid test kit through the saliva using, which is what we call a lateral flow assay. And of course, there are also concerns of looking into several COVID genes, okay, in a single multiplex PCR assay molecular diagnostics. So those are molecular methods which are good, okay, so which are the standard. Okay, but what we complement is in the next slide is we use artificial intelligence and Raman spectroscopy so to work into looking into the pattern recognition itself. Okay, so in the next slide, when we talk about optical spectroscopy, which is in the next slide, okay, so it's looking into UV or infrared radiation, right? So that UV and IR is good, okay, but they are sort of destructive, okay? So it means that we know that UV and IR will break DNA, will change the chemical bonding and sort of things, okay? So those are destructive. So when we do a Raman spectroscopy, okay, it's looking into the light scattering itself, okay? So we have a sample, okay? And then we put it at incidence light, okay, and then we record the scattered light. Okay, so there are examples in the next slide, which those that what we see in this slide is actually the patterns. Okay, the blue, red, green, right? So those are different patterns, right? So looking into the light scattering and the patterns of it, okay, so then studies have characterized T varieties, okay, so they use it and then they use it to look into dengue virus, okay, to differentiate what are the positive dengue virus and what are the negative dengue viruses. Okay, so in the next slide is why we need pattern recognition is because there are problems where in my next slide, it is for both storage, okay, of the biosample itself, right? The viral transport medium. Okay, so that is a sample that uh, clinician use, okay, to isolate RNA and then reverse transcribe and do real-time PCR to see whether it is positive or negative viruses. So those are good and they are good, the gold standard. One of it is what we do now and what we had published is because we do optical methods. Okay, so that is very straightforward. So the next slide actually shows that where is our sample? Okay, it's in the cuvette. Okay, and we have the coupled charge device. Okay, we have a monochromator, we have the Raman scattered light, we have the long pass, and then we have the laser diode, okay, which is the source, okay, and the Raman and the which has been scattered light, and then that will be recorded into the system. Okay, so when the system recorded, okay, so in my next slide, we do positive COVID samples and negative COVID samples. So when we talk about the samples, what we did is we use viral transport medium, VTM. Okay, so we see that there are patterns. Okay, so those peaks that are highlighted in yellow, so those are actually is used to be present in positive samples only. Okay, so the peaks of different wavelength in terms of cm. Okay, so and then the relative intensity. Okay, so those are the highlighted peaks that we use. So we can see that in this slide, okay, the Raman spectroscopy of the positive sample, and the next slide, which is the Raman spectra for the negative sample. So they have different peaks. Okay, so that leads us to know that, okay, when we have this pattern, this particular peak, it is positive. When we have another pattern, so that is negative samples, okay? So we have shown that this study have a sensitivity and specificity of more than 95%, okay? So in conclusion of the next slide, okay, so we have shown that the Raman spectroscopy is rapid, okay, it's highly sensitive method. So we can call the sample as positive and negative in 5 seconds, okay. So if you do a conventional DNA isolation method, and then you need to isolate the DNA, RNA, okay, for this context is the RNA, and then you need to do either one-step reverse transcription, real-time PCR, or two-step, depending on the time and the budget. Okay, because we know that molecular testing is expensive, and they are really expensive. So that's why cost is another issue also for certain countries. So that's why Raman spectroscopy is rapid, sensitive, okay, and is cost effective. So what's next is we can use it for looking into COVID, right? So this Raman spectroscopy actually can look into as well different diseases, not only COVID viruses. Okay, it can look into maybe sputum samples, okay, from a sepsis patient. It can look into maybe for blood, plasma, or serum. Okay, that there is a particular disease that people would like to detect. So can be done by using Raman spectroscopy. So the application of Raman spectroscopy and pattern recognition is a lot. Okay, depending on what are the targets that we want, okay, then we can cater to it. Okay, but the challenges of Raman spectroscopy is an predictive algorithm or pattern recognition algorithm is because we need a lot of data, a lot, like thousands or tens of thousands of data to develop that algorithm. Right, like what we have in artificial intelligence currently. So we are using it in the context of language learning model, right? So, but we can use the same concept to disease detection as well. So in my next slide, okay, so when we talk about SDGs and then what we are currently doing, right? So the work that biotech currently doing has an impact towards how we form the policy. So in Malaysia, we have the National Biotechnology Policy 2.0, okay, and we have a few policies that are related to biotech. So for example, the biosafety, the medical research ethics approval. So those actually are the policies that has been designed from the input of biotech itself. So, and also those policies actually, and in terms of research grant policies now are encouraging industry, academia, collaboration. So, and as well as policymakers. So, we have actually got a UKRI grant, okay? So through my pair, so that is looking into urinary tract infection. So is more towards how we want to use the technology that we have, okay, through collaboration of many countries, okay. We have from the UK, Queen Mary, okay, University of London, and then we have partners with that project. We have someone from Vietnam, we have someone from Bangkok, I think Philippines as well is in the project. So in that project is looking into urinary tract infection. Okay. So it's looking into more towards a policy level. Of course, that's where biotech comes in because for a policy to be developed, you need data. So you need data on top of what are the disease incidence and prevalence rate. You need data on what are the tools that can be treated clinically and using biotech tools as well. Molecular, non-molecular tools. Okay. So in my next slide, I will talk about what I did, okay, which is with collaboration of clinicians, okay, the doctors, the surgeons in the hospital itself, focusing on colorectal cancer. So in just worldwide cancer incidence, okay, is more than 20 million. And then we look at the worldwide cancer mortality is approximately 10 million, right? So this is, I pull it for the IARC WHO recent statistics. Okay, so we can look at we have lung, we have breast, colorectal. So I apologize if it is too small. Okay, prostate and stomach cancer. Okay, so then when we look into the next slide, which is looking into Malaysia. Okay, in Malaysia itself, we have approximately 50,000 incidents of cancer cases overall, and then the mortality is approximately 30,000, right? So in the incidence, so breast is the highest, followed by colorectal. But in the mortality, lung is the highest, followed by colorectal, and then breast, right? So this actually shows us that yes, we can detect a lot of breast cancer. There are a lot of breast cancer, but the mortality is low, right? Why is it that? It's because through early detection. So breast cancer is easily screened, and there are a lot of programs for breast screening in Malaysia itself. So it's ongoing throughout the years. There are many screening programs. And then, and in terms of the technology, breast cancer can be easily screened using mammograms. Okay, so looking to that, and we compare the breast and the lung, right? So lung actually has the lowest incidence, okay, but the mortality of lung cancer is the highest, right? Because usually when lung cancer is detected, patient have cough blood and it's already stage 3 or stage 4, where the cancer has already metastasized. So focusing on colorectal cancer, okay, is the second. So a standard colorectal cancer screening is through colonoscopy. Okay, put a camera inside the colon and inspect. So if there are polyps and then or if there are abnormal growth, then they will take the tissue sample and through screening, right? So because of these incidence and mortality cases in colorectal cancer, okay. So in my next slide, so actually I'm citing a Nature Genetics article where if the cancer is only 0.01 cm, okay, clinically, okay, is undetectable because 0.01 is very small, right? So that actually is can be metastasized. So means that the cancer has already migrated to another part of the body, even though it is 0.01 cm. So they observe it is in 81%, okay, of their samples, okay, which is 17 out of 21 patients, okay. Even though the cancer is 0.01 cm, it's already metastasized. So this shows that even though there are colonoscopy and things like that, right? But the technologies could not detect 0.01 cm. We can't even see. Okay, it's very small. And cancer, of course, there are two main types. One is sporadic and then one is familial. So sporadic is through the accumulation of mutation. Okay. So maybe early on there's APC mutation, KRAS mutation, SMAD2, 4, and then P53. And if there are more other mutations, then the cancer would undergoes metastasis, would migrate to different places. So in familial is mostly inherited, okay, colorectal cancer through inheritance. So what we did was in my next slide, so it's a method of summarizing. We do metabolomics, we do molecular, and we do next generation sequencing. Okay, so metabolomics, we look at what are the metabolites, okay, and then we do what are the gene expression that has been expressed. And then we look into what are the gut microbiome itself. Okay, so in my next slide is I'm showing that what we inside the operation room, okay, me and the clinicians, okay, inside the operation room, okay, looking at the patients, okay, collect the tissue samples, okay, itself, okay. So working with the clinicians side by side, okay, to see a particular case and then, okay. So because in biotech, we always see things very small, right? So that's why when me go into the operation theater, so I see that, oh, that is the big sample, okay, the entire tissue sample. So in my next slide is actually the data from our gut microbiome, okay. So when we compare the cancer tissue and the healthy tissues, so we see that there are more protobacteria in the cancer tissue, okay, and there are reduced Firmicutes, Bacteroidetes, and Actinobacteria in the cancer tissue, okay, because for example, Firmicutes is they produce short chain fatty acids, right? So it's actually can reduce the oxidation and inhibit the nitrate producing gene. Okay, so that's why it protects the normal tissue from being cancer. Okay, Firmicutes. So then we found out that there's one particular genus which is the Campylobacter genus. Okay, which is significantly high in cancer patients. So in my next slides is we look into what are the KEGG pathway, okay, and then what are the normal possible metabolic pathway, okay. So what we found out is that there is a bile acid related pathway, which is the FXR receptor. Okay, so then of course FXR actually can be activated by the bile acids. Okay, so and we look into what are the phylum or genus that contributes to that bile acid pathway. Okay, so and then we further on do a metabolomic study in my next slide, which we compare the polar and the non-polar extracts, looking into what are the chemicals that is increased. Okay, so we found two main things. Okay, one is glycerophospholipids, which is a cellular component, and then one is the carnitine system. So the carnitine system actually is for beta oxidation or fatty acid oxidation, which produces energy. So we know that when cells divide, they need energy, right? So that's why in cancer, so this carnitine system actually is higher. Okay. And then in my next slide actually is summarizes the pathway that we found, okay, including gene expression analysis, okay, which I did not show the data, but it summarizes here. So we found that from the left to the right, okay, from the left we see that there's increased Campylobacter genus. So then together with Campylobacter genus, so it actually reduces activation of FXR receptor. Okay, and then moving forward, it activates a lot of glycerophospholipid related pathway, and then and then the fibroblast growth factor increases, right? So that actually leads to the activation of fatty acid oxidation for energy production. So that's why we actually this study hypothesized that in the mitochondria itself, so when energy is being produced, so there are several pathways. So one of it is through the carnitine system, okay, which is what we know already, right? So, but in our case, okay, we find the carnitine gene is downregulated, means that the carnitine system is not the main contributor to produce energy in the form of ATP. So then we find fibroblast growth factor FGF19, okay, so is upregulated. So this shows that FGF19 gene, okay, is the alternative pathway to activate the mitochondria to undergo fatty acid oxidation for energy production. So that's why in the next slide, okay, so we actually look into biotech as a whole. So we look into what are the careers, what are collaborations. So actually it's require multidisciplinary and interdisciplinary skill, whether it's molecular biology, whether it is genetics, whether it is working with the doctors, whether it is working with the bioinformatician, right? So by working with different people or different fields, okay, we can see that looking into biotechnology and its contribution to health, so we need a multidisciplinary approach. So people must talk to each other, work together. So of course, in my next slide, which is the conclusion, so we need to use biotechnology, okay, to of course, for better healthcare, whether it is through emergency care or through pharmacy services. So in my next slide is all right. Thank you.
Okay, thank you so much, Dr. Lucky, for the outstanding presentation. I was amazed to learn just how far biotechnology has come in advancing human well-being, especially in achieving the Sustainable Development Goals. Before we proceed to the Q&A session, allow me to share a brief summary of the main discussions and key takeaways from this session. This presentation highlights biotechnology as a strategic tool to develop practical solutions, particularly addressing the limitations of current detection methods, which remain constrained by time, complexity, and equipment requirements. To overcome these barriers, a biotechnological approach utilizing a QD-based FRET assay was developed, offering high sensitivity, reliable performance on clinical samples, and accurate target gene identification. This innovation represents a promising step toward faster, simpler, and more precise diagnostic methods for real-time clinical application. Moreover, broader global health challenges such as infectious disease and pandemics further emphasize the critical role of rapid testing in achieving timely and effective healthcare responses. It also highlighted how molecular diagnostics have advanced healthcare, contributing to technologies such as next-generation sequencing, CRISPR-based diagnostic, and personalized medicine. In recent years, during the COVID-19 pandemic, molecular diagnostic played a key role in the rapid sequencing of viral genomes. We had the chance to see just how far biotechnology has advanced in innovating for better lives. Now, we move to the Q&A session. For any participants who wishes to ask, you may use the raise your hand feature, or please type your question in the chat room. Right now, we actually already has one question on the chat box from Puspa. I'm very interested in your approach to combining multi-omics data for cancer research. From your experience, what's the most effective way to integrate datasets like transcriptomics and metabolomics when they come from different platforms or scales? Okay.
Okay. So, yes, I understand that is one of the big challenges that we have for actually data analytics. So most importantly, what we look into now is big data analysis, where everything is in the cloud. When you go to NCBI website, you can search the gene, right? Thousands of genes. You can search the genome, okay? Genome of I don't know, all the species that has been sequenced is inside. So because they comes in different format, okay, different scale, okay. So that's why I think that the most effective way I would suggest is that to look into maybe is there any bias, okay. So perhaps the data from metabolomics comes from Asian, and then you look into the data of transcriptomics from European. So it's difficult for you to connect because genetically Asian and European are different. So that's why, and our diets, our lifestyle are different. So that's why the metabolomics and all the transcriptomics for two different regions so might not be representable. So that's why just don't introduce too many biases inside. Yeah. So, but they are complementary data. So that's why some data, for example, next generation sequencing data. So they comes in different formats, right? And from different platforms. So as well as metabolomics also. So if you use different brands or different companies, they will comes in different formats. Then you need to transform the data, you need to change the data through a common format. So for metabolomics is MZXML file format. So, and for next generation sequencing is more towards maybe FASTA or FASTA-Q file format. Yeah.
Okay, I hope that answers the question. Okay. For the next question, we had from Salman Jafar. How can health biotechnology innovations contribute to achieving the sustainable development goals of good health and well-being?
So, when we talk about good health and well-being, so it is in different contexts. So it can be preventive, it can be diagnostic, or it can be a treatment, or it can be just improving the quality of life, right? So then the innovation of biotech is encompasses this field, right? So it can be applied to this field. So from prevention, what biotech can do is for maybe early screening of certain genetic mutations, okay, of the child, okay. So that is actually where we look into, I'm not sure about Indonesia, but in Malaysia, Sabah, North Borneo specifically, we have a lot of thalassemia and hemophilia. So those are actually inherited. So maybe what the clinics are doing now is to screen the patients, screen the parents. So if they are high risk of bearing a child with that thalassemia or hemophilia, then they will go to genetic counseling. Yeah. So it contributes bits by bits. I would not say that it is like we have one solution for everything, right? So it's difficult. So that's why like we have different fields of biotech as well. So that's why all these natural product, genetics, metabolomics, bioinformatics, biochemistry. So those actually contributes through the SDG of good health and well-being in different ways.
Okay, I hope that answers your question, Salman. We have more questions. This time is from Anne Farhana. Given the benefits of Raman spectroscopy, such as shorter time and simpler way for disease detection, what is your opinion on the potential for future applications of this technology in hospital settings?
So with Raman spectroscopy, yes, it is a shorter time, but the challenges would be to develop a small point of care testing device, okay, that we can just, it's like a glucose meter, right? So like nowadays, to test the glucose is very easy, right? You just go to the pharmacy, buy the device, and there's a biosensor, and then just to check, right? So so Raman is the challenge is to develop a device to record the Raman spectroscopy data and to analyze it and to update the algorithm from time to time. So it requires a longer time, okay, very, very long, okay, maybe more years to go, okay, because currently the Raman spectroscopy we have is on the bench, okay, it's not as per se really big, but you would still need someone to carry and then someone to turn on the laptop, turn on the device, and yeah. So it still requires some time to make the model smaller, so it is more easily to carry.
That's another answer for Anne Farhana, if that's enough. Okay. For the next question, we have from M. Umar Azif. Your research about oncology is interesting. I want to ask about the tools Dr. Lucky used for transcriptomics, especially for RNA sequencing analysis.
All right. So when we talk about RNA sequencing, right, transcriptomics, right? I know that it is easy for us to have many data, right? To generate a lot of data, right? But then how you want to analyze the data, right? That is the main challenge actually. So it's easy to generate a lot of metabolomics, transcriptomics, genome data, but then which one out of the how many gigs of the data that we produce, so which data actually is the quality data, which actually is the data that we want, right? We want to select them, we want to have a quality control, QC them, right? So um, for the questions is focusing on transcriptomics per se. So in transcriptomics, it's a lot, right? So you can use most widely is Illumina platform, right? So that actually Illumina support is really good. Okay, so they have online analysis software and then things like that. So, but I understand it does comes with limitation, right? So unless I'm not sure how Undip's bioinformatics programs and teams develop. So here, unless you can have a very good computing skills, you know Python, you know C++, you know how to use RStudio, okay, you know how to use Linux, okay, that is offline, so you can do it by yourself, everything, right? So then if you can't do that, then I would recommend you to work with someone who can do, or you can use Illumina online analysis. So of course, there are limitations on that as well, because sometimes we want more advanced, right? A nice pie chart, a nice diagram, right? So in fact, the diagram that you see in my gut microbiome one, that is generated using RStudio. Okay, so that's why it's not generated using Excel, Words, or Paint, or Photoshop, things like that. So it's generated using RStudio. So when I generated that, yes, it's difficult, but then I do not say it is not possible for biotech to learn computing skills, because it's easy. Okay, so when you learn the first step, it's already gone through, then the rest of it is easy. When you understand the fundamental concept of computing, what are the input, output data, and yeah, there are a lot more lah. Okay, so because it's like we learn about genetics, we learn about PCR, it's the same, just a different field. Yeah, so if you're looking for RNA sequencing analysis tools, okay, so I would say that make sure your RNA sequencing method workflow is correct. How you store your RNA, how you isolate your RNA, how you do your library preparation, and then when you put into the Illumina platform or whatever platform, then make sure that the library is being QC'd, the instrument is working well, the flow cell is good, and then the adapters, the light, all the linkers are good that you use is correct. Yeah. So then you have a lot of data. How you want to analyze is either you do it yourself manually or you use the online tools, that a lot actually, not only Illumina. In fact, if you go to galaxy.org, I think. Okay, so they have a lot of online tools platform. Just that you need to learn like how to click, what is the file format, macam tu.
Thank you, Dr. Lucky, for the answer. I hope to Umar that was answer enough. For the next question, we had from Tasrari Colby. On your slide, you have put the future research will use AI. What kind of AI did you use on the research? Because I think it's very interesting that the use of AI on bioinformatics. How is AI developing for bioinformatics in Malaysia?
So when we talk about artificial intelligence, actually it comes from 3 years back, which is the first ChatGPT, right? So then everything is on AI going onwards, right? So whether you are looking a language learning model or whether you are looking at pattern recognition, right? But in the context of using AI for bioinformatics, so it's actually the one that I shared just now where we do the protein and then the molecular docking and everything, okay? So those you can actually automate. So when we talk about automation means I have this PDB file. A PDB file is a protein file, okay? So it comes from the PDB database. Okay, I have this file. I have this list of chemical compounds. So I just press a start button and then it will tell me what is the potential good compound. Okay, so in terms of AI policies in Malaysia, so currently the Ministry of Higher Education is looking into what subjects is integrating AI. So in fact, in the biotechnology program itself in Universiti Malaysia Sabah, so we have a bioinformatics program, and inside we incorporate the components of artificial intelligence, where we look into what are the probability of a particular patient's having high blood pressure based on the blood profile, right? Because when people go for blood testing and blood checking, they will have the full blood count and then cholesterol, things like that, right? So then with that data, okay, we compile it together and using the same pattern recognition concept, okay, we look at the pattern of diabetic and the pattern of healthy. Then from there, we look into a blind test subjects, okay? Having this profile, is it categorized in diabetic or is it categorized in healthy? So that is what we look into it.
That was a nice answer. I think we still have time for the last question from Nisa. I've seen a rapid test kit for CRC. Given that CRC involves multi-step molecular and microbiome changes, do you think a single marker rapid test will ever be sufficient, or should we be thinking in terms of integrated signatures even at the point of care test?
So I think that there is no one marker for all diseases, okay, especially for CRC, because CRC can develop at different stages and the gross pathology of the CRC is very different. So we need multiple markers. One marker is not enough to tell that what is the stage of colorectal cancer and then what are the maybe the physiology of the cancer tumor itself. So we need several markers, because if you use one, how sure are we that that is the one, right? So that's why we need multiple to sort of a confirmation and to support each other of the different markers for colorectal cancer.
Okay, thank you so much to Dr. Lucky and the audience for the amazing discussion time. Unfortunately, we've come to the end of our question and answer time. Before we officially close this session, we would like to present an appreciation to Dr. Lucky for his contribution to today's webinar session, followed by a quick documentation moment. We invite Ms. Arina to come and present the certificate of appreciation to our distinguished speaker, Dr. Lucky.
Hello, Dr. Lucky. It's very nice to see you again. And I would like to deliver my gratitude for your willingness to present today, even on the weekend, but you still make time to be here with us. And we truly appreciate for your amazing presentation about the health biotechnology. And also, we would like to express our sincere gratitude to OMS, because this collaboration, you are very welcoming, supportive, and helping us to make this event be success. So here is a form of our appreciation for you, the certificate. Thank you again. Thank you so much, and thank you so much for OMS to want to collaborate with us. Thank you.
Okay, we will take a quick documentation to commemorate this moment. I hope for all audience to turn on the camera and join us in this documentation. Okay, I will count. 3, 2, 1. 3, 2, 1. Okay. Dr. Lucky, the original digital certificate will be sent later. Okay. On behalf of the committee, I would like to express our sincere gratitude to Dr. Lucky Gopohwa, PhD, for the insightful and inspiring presentation. Everyone, please give a round of applause to our distinguished speaker. Okay.
Thank you.
Thank you so much, Dr. Lucky.
Yeah, thank you, Miss Arena.
To Dr. Lucky, if you have any other agenda after this, you may kindly leave the Zoom room. Thank you very much. Okay, that was truly a fun webinar event. We had two speakers extremely skilled in their field to share their expertise and share their knowledge to us audience. Now, before we end this webinar session, let's take a short break and enjoy a light ice-breaking session to refresh our minds and keep the energy flowing. But before that, I would like to remind for all participants to fill in the attendance form, the links on the chat room below. Okay. For this ice breaking, we will just do a simple quiz game on a platform called Wayground. For some people may know this previously as quizzes. You may easily access the link shared on the Zoom chat or on the screen by the operator. Or even you may take a look at your WhatsApp group to access the QR code. Okay. We will give time for the audience to join in the Wayground, and we will start at 11:03. Okay. The links are already sent on the chat. If you win the games, there will be a prize. So be sure to not miss this one out. Okay, right now there's only 10 participants. We have around 100 participants here. Come on, guys. Let's join in the games. And who knows, maybe you will win a prize. So not only you finish this webinar event by knowledge, but we also have prize in your hands. Okay. Still have 2 minutes left for participants who wishes to join. The QR code is already presented on screen. You may easily scan the QR code on your camera or just type in the wayground.com and fill in the code below. We have 24 participants right now. One more minute, guys. One more minute before we start the game. It's just a simple quiz game. Simple enough to refresh our minds, loosen up our muscles, and enjoy it. Okay, 31 participants. Ooh. 36. One more minute. 37. Can we get into 50 before we start? 38. 39. Okay. 40, 10 more. I think we can get 10 more before we start the game. Come on, participants. Okay, it's okay. We will still give time for those who wants to participate in this game. We will wait until 50? 50 participants. I think we can get into 50. Okay, 6 more. Five more, guys, or 5 more. We will start in 1 more minute. Okay. 47, 48, 49, and 50. Who will be the 50th participants? Don't hesitate to join in the game. It's a harmless game, just a fun one to lighten up the mood, lighten up the session before we officially close this one. Okay, we still have more time for 3 more people to at least join in to make it 50. I think 50 will be a great number. 48, 49, 50, 50. Who will be the 50th person? Ooh, 49. One more person. Oh, it's already 11:05. Okay. Okay. Okay, we will, because the time is already 11 past 5, 5 past 11. We will now start the game in 3, 2, 1. Oh, 50. How many member states does ASEAN have? Oh, wow, that's ASEAN. ASEAN is short for Association of Southeast Asian Nation. Anyone who remembers the social? Okay, the first one is a person named First and followed by Adeline. Okay, keep it going. 15, 14, 13, 12, 11, 9, 8, 7. Ooh. 3, 2. Oh, I get other answer by 45%. ASEAN has 10 member states. For this next question, if you could have a superpower to help create a sustainable world, which would be the most impactful? Eliminate plastic waste with a snap of your fingers, plant 1 million trees in 1 minute, provide free renewable energy for everyone, or end global poverty overnight. Keep it coming, guys. The answers in 10. We still have 10 seconds left. 34 from 52. If you could have a superpower. Whoa. Okay. Next question. Which combination of the SDGs is most directly related to cross-border? We have SDG 3 and 6, which is health and water, industry and partnerships, hunger and oceans, education and land. You still have time to think about the answers. Okay. We have again first Adeline and Adnan. Ooh, a new member. Oh, the answer is the industry and partnerships. A startup creates an app for farmers to sell crops directly to consumers. Which SDG is most related? A startup creates an app for farmers. Is it no poverty? Is it zero hunger? Or is it industry, innovation, and infrastructure? Or maybe is it decent work and economic growth? Okay, we have 6, 5, 4, 3, 2, and 1. We have 43 answers. It's decent work and economic growth. For anyone who paces with time, may be the winner for this game. And the next question is, in 2025, the Aspire competition is a collaboration between universities from which two countries? Oh, this is a very easy question. I think this is a very easy question. If you paid attention from the first, you would know. Okay, we have first an Adeline again. 39. Keep it coming. 8, 7, 6, 5, 4, 3, 2, 1. Okay. How much more questions do we have? Question number 6. If Earth could speak in 2025, what would it be the most urgently shout? Is it stop cutting my trees? Or I need clean water? Give me fresh air. And can you chill with the CO2? Two and one. Okay. Next question, still related with the SDGs, same as our theme for today. Which sustainable development goals is represented by these emojis? I think we have a leaf emoji, a factory, and a recycle. Ooh. What emojis would these represent? 9, 5, 4, 3, 2, and 1. 42 answers. Oh, it's SDGs number 12. I answered that wrong as well. Which SDG is promoted by refusing a single-use plastic bag? Is it sustainable cities and communities, life on land, responsible consumption and production, or is it life below water? If you answer the question quickly, you may have a chance to win the game and possibly receive a prize. So everyone, please join in on the games. Who knows, maybe you will get lucky and win a game. Question number 9. If the SDGs were a music group, which one would be the official leader, without which the group would fall apart? If there's a leader, what do you think would be the magne or the visual? Okay. Still have more time for the questions, for the answers to come. Let's go. And 1. Oh, it's the partnership between global. That is true. The 10th question, which sustainable development goals directly addresses the reduction of global CO emission? They're affordable and clean energy, responsible consumption and production, climate action, and life on land. Which SDGs directly addresses the reduction of carbon dioxide emissions? Is it affordable or clean energy, responsible consumption and production, or is it climate action, or is it life on land? And that is the end of this icebreaking session. Thank you so much for the participants who have joined in, and congratulations to the winner. Ooh. Oh, congratulations to everyone. Okay, the first winner. I think. Oh, okay. Congratulations to Zulaika, to Silviana, to Adeline Hartono for winning the game. That's really cool. Well, it seems that these top three people are very knowledgeable on the SDGs in spirit of this webinar event. To those who want, to Zulaika, Silvania, and Adeline Hartono, don't forget to personal chat to the CP, which is Colby, to confirm. And who knows that you might get a present, and don't forget to screenshot it first as proof. Okay. Okay, now that the icebreaking session has ended, we have now reached the conclusion of today's webinar session. It has been an honor to learn how biotechnology contributes to achieving the Sustainable Development Goals. To all participants, thank you so much for your active participation and enthusiasm throughout this session. We hope that this session has not only broadened our understanding, but also inspired the younger generation to actively take part in building a sustainable future. With that, allow me to wrap up this webinar session. It's been a pleasure for me to moderate this session. I will now return the next session back to our Master of Ceremony, Almas. Thank you so much and see you on the next occasion.
Okay. Thank you so much to Amy for moderating the webinar. So, how do you feel after the moderating our two amazing speakers? Since you're also a biotechnology student, I'm sure the topic will be very relevant to you.
Oh, it's a very it's a very great experience because I can hear more, I can gain more knowledge from two experts who are the leading expert on their field, which is very interesting. And it's not just every day I get to moderate two very, very amazing speakers. So yes.
Okay, so yeah, since you're also a biotechnology student, I've said that earlier that the topic will be very relatable to you. So yeah, do you interested in biotechnology marine or not? Since we're talk about biotechnology marine earlier.
Uh I'm interested in both area, which is the marine and the red biotechnology. Healthcare is also a very interesting topic, and marine as well, considering that we are in Indonesia. Our land is surrounded by oceans. We have massive biodiversity. It's really eye-opening to see just how biotechnology, the study that I'm studying right now, has a lot of potential in building a sustainable future for the younger generations and more.
Yeah, I agree. Biotechnology marine is indeed interesting and also very many things to learn in it. So I guess since you're also still a second year in biotechnology, I think you still had very much time to think to which biotechnology you want to more get joining to. Okay. At this time, I would like to say thank you to you for once again. Thank you for being such a magnificent moderator, and thank you for moderating the webinar very well. At this time, I would love to present a certificate of appreciation to our moderator, Aimee, for their excellent guidance and support throughout today's session. Okay, so now we would like to invite Ms. Adeline for giving out the certificate.
Okay, thank you, Aimee, to moderate today's event.
Thank you so much for having me here. It's truly an honor for me.
Okay, now we will take a moment to capture some photos with our moderator. Let's just get that into it. Please turn on your camera so we can capture this memorable moment together. Okay, I'm going to count it down. In 3, 2, 1, pose. Okay, another one, maybe with Aspire sign. Okay, I'm going to count it down in 3, 2, 1. Okay, thank you very much for Miss Adeline and Aimee. Okay, thanks. Once again, I'm going to say thank you to the moderator for your valuable contribution. At this point, the moderator will leave the meeting. We appreciate your time and effort. Thank you, Aimee.
Everyone.
Okay, okay, I'm going to say thank you to Aimee for once again. Oh yeah, and since the webinar session has concluded, participants who have other activities can leave the event, but for the competition, participants are expected to stay until the end of the event because after this is the awarding session for the Aspire 2025 competition. Oh, also, don't forget to fill in the attendance form so you guys can get the certification. It's now time for lunch break, so you guys can pray, eat, and take some quick rest. And we'll begin again the next session at 12 past 10 p.m. Thank you.
Sometimes, a tiny stuff or things out of sight might turn to be main cause of every possible tragedy on Earth. Hence, a living creature should be observed for the sake of science.
Selamat datang di Fakultas Sains dan Matematika Universitas Diponegoro. Kami berkomitmen menjadi fakultas riset unggul berstandar internasional, mendukung Universitas Diponegoro bermartabat dan bermanfaat, serta mewujudkan World Class University ranking 500 dunia, dan mencetak lulusan berdaya saing global serta nilai moral dan etika yang kuat.
Accordingly, we put many scientists out, dedicated every single piece of our heart for each research. Assisted with sophisticated tools and advanced technology, no matter how complicated the situation is, it will be elaborated to bring out the solution. Compounds were studied, collected up to form a theory. Every single invention were integrated with another invention.
Melalui integrasi yang harmonis antara pendidikan, penelitian, pengabdian, dan kemahasiswaan, kami berkomitmen mencapai standar World Class University dengan keunggulan yang terukur dan berkelanjutan. Kami membangun ekosistem pembelajaran yang holistik, mengintegrasikan pengembangan akademik, keterampilan soft skill, dan hard skill. Penelitian dan pengabdian kami terpusat pada pengembangan solusi inovatif yang strategis dan berkontribusi nyata bagi masyarakat seperti mengentaskan kemiskinan, memperkuat ketahanan pangan, sekaligus mencetak generasi pemimpin yang visioner, berintegritas, dan berdaya saing global.
Giving wide possibilities for new circumstances to be explored, investigating the source and resources to find out novelty, and proceeding a sequence of practicums. Cultivating collaboration, domestically as well as abroad.
Ini adalah unit produksi produk inovasi yang dihasilkan oleh Universitas Diponegoro dari Fakultas Sains dan Matematika yang berbasis teknologi plasma. Ada dua produk yang sudah dilisensikan oleh Universitas Diponegoro ke PT Dipo Teknologi, Zeta Green dan D-Ozone.
Considering the wide range of science, therefore, having strong mutual relationship with as many institutions as possible is the key for any kinds of successful research.
SEMEPSA, Research Center ini terbuka lebar bagi semua pihak ya, yang ingin mengembangkan riset-riset yang muaranya kepada pengembangan aplikasi biomonitoring, maupun juga pengembangan sustainable aquaculture. Kita sangat membuka lebar untuk kerjasama.
Anything impossible turned to be possible. Change something insignificant into something worthy. What was only a plan transformed to be a considerable execution. Upon every lecture, whether in the classroom or in laboratory, students were driven in an embedded discussion,
selama perkuliahan di sini aku ngerasa dapat banyak banget ilmu, dapat banyak banget pembelajaran, mulai dari pembelajaran di kelas bersama dosen, dari sisi teorinya, tapi di sisi lain, dari sisi praktikalnya, aku juga dapat banyak banget ilmu melalui kegiatan praktikum yang menurut aku pribadi sangat baik sekali untuk menunjang aku di dunia kerja.
Ketika aku masuk ke Smart Building ini, pelayanan yang aku dapatkan sangat baik karena staf yang melayani sangat ramah dan siap membantu apa yang kita butuhkan. Di samping itu juga, fasilitasnya sangat memudahkan kita, seperti pintu yang dapat terbuka secara otomatis dan lift yang dilengkapi dengan sistem sensor. Terdapat juga ruangan yang cukup luas dan nyaman untuk mengikuti kuliah umum.
Supported with adequate facilities, the Faculty of Science and Math Diponegoro University adhere to inclusive education.
Di perpustakaan ini, aku mendapat berbagai macam fasilitas, yaitu ruang belajar yang nyaman. Selain itu juga, aku dapat dengan mudah mengakses media pembelajaran, seperti buku, jurnal, karya tulis ilmiah, yang dapat memperluas wawasan dan juga membantu aku dalam pengerjaan tugas aku.
Proved by providing enrollment for disabilities. Its students deserve the same opportunity to create their own bright future.
Dengan strategi peningkatan kualitas mahasiswa, optimalisasi fasilitas, dan pengembangan sumber daya manusia, kami menerapkan tata kelola efisien dan akuntabel, didukung teknologi informasi terintegrasi untuk memperkuat keunggulan institusi. Melalui kolaborasi internasional strategis dengan universitas terkemuka dunia, serta sektor industri, Fakultas Sains dan Matematika Universitas Diponegoro siap menjawab tantangan global, memberikan solusi berkelanjutan, dan berkontribusi pada masa depan Undip yang bermartabat dan bermanfaat.
We strongly believe that all the dream come true emerge from the soul that keeps vibrantly enthusiastic.
Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera bagi kita semua. Atas nama Sivitas Akademika dan seluruh warga Fakultas Sains dan Matematika, saya Profesor Dr. Widowati, Dekan Fakultas Sains dan Matematika Universitas Diponegoro, mengucapkan selamat atas diterimanya saudara-saudari di Fakultas Sains dan Matematika.
Assalamualaikum warahmatullahi wabarakatuh. Program Studi Bioteknologi Universitas Diponegoro berdiri pada tahun 2018. Program Studi Bioteknologi memiliki visi pada tahun 2025 menjadi pusat pendidikan, penelitian bidang bioteknologi sumber daya hayati yang berkontribusi dalam skala nasional dan internasional dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan. Untuk selanjutnya, misi dari program studi bioteknologi Universitas Diponegoro terdiri dari 1. Melaksanakan pendidikan tinggi jenjang sarjana bidang bioteknologi sumber daya hayati yang unggul, bermoral, beretika, berkompeten, berkarakter, dan berwawasan kebangsaan. Misi yang kedua adalah melaksanakan dan meningkatkan kualitas penelitian yang kreatif dan inovatif dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan berdasarkan prinsip-prinsip bioteknologi. Misi yang ketiga adalah melaksanakan dan meningkatkan kualitas dan kuantitas pengabdian kepada masyarakat melalui pendidikan tentang pemanfaatan dan pengembangan sumber daya hayati yang berkelanjutan. Bioteknologi Universitas Diponegoro berdiri sejak tahun 2018. Setelah 4 tahun, kami telah meluluskan sekitar 25% dari angkatan pertama. Dan dari lulusan program studi bioteknologi tersebut, ada beberapa mahasiswa lulusan kami yang telah memperoleh pekerjaan sebelum lulus. Kemudian yang membuat kami berbahagia adalah dari lulusan tersebut juga memperoleh penghasilan yang jauh di atas upah minimum rata-rata.
Hello everyone, my name is Nabila Maharani and I am a fresh graduate from Diponegoro University majoring biotechnology study program. I was pursuing this study for 3 years and 10 months and Alhamdulillah today I am graduating from Diponegoro University. When I was a student in here, I got a lot of experience such as laboratory experience, field sampling experience, and one cool experience that I got is that I have a chance to do a research collab with a university overseas, such as Boston University. And with all of the experience that I got, I am optimistic that I could get a job opportunity in a research and development field. And do you know that this biotechnology study program has a really great laboratory facility that you could do all of your research in here.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Muhammad Iskandar Zulkarnain, alumni dari program studi bioteknologi Universitas Diponegoro angkatan 2018. Pada program studi bioteknologi, saya telah mempelajari dan mengalami berbagai bidang riset, diantaranya adalah genetika, mikrobiologi, teknologi fermentasi, dan kultur jaringan. Saya yakin dengan pengalaman yang saya peroleh, saya dapat berkontribusi di industri hijau Indonesia. Bioteknologi keren!
Halo semuanya, perkenalkan saya Roma Redion, saya merupakan alumni program studi bioteknologi angkatan 2018 dan hari ini puji Tuhan saya telah diwisuda di tahun 2022 dengan predikat cum laude dan juga mendapatkan penghargaan dari dekan dengan meraih IPK tertinggi di atas 3,9. Selama saya berkuliah di bioteknologi, saya mendapatkan berbagai pembelajaran, terutama dari tenaga pengajar, dosen, bahkan dari kakak kelas kami juga. Dan ketika kita berbicara berkaitan dengan bioteknologi, di mana bioteknologi merupakan suatu ilmu yang mempelajari tentang pemanfaatan agen hayati untuk kepentingan barang dan jasa, saya sangat yakin bahwa bioteknologi ini merupakan ilmu yang sangat luas, ilmu yang mengglobal. Hal ini juga dibuktikan bahwa bahkan sebelum saya lulus dari bioteknologi itu sendiri, saya sudah mendapatkan pekerjaan, yaitu di Staff Research and Science di PT Haldin Pasifik Semesta. Berdasarkan hal tersebut, saya juga berharap bahwa bioteknologi juga bisa mencetak berbagai sarjana yang mampu kompetitif untuk mendapatkan pekerjaan yang terutama di bidang bioteknologi juga. Bioteknologi keren.
Saya Masda Manurung, ibunda dari wisudawan Romario Dion yang merupakan alumni dari program studi bioteknologi Universitas Diponegoro. Saya sebagai ibunda merasa bangga karena berkat berkuliah di bioteknologi, anak saya, Romario Dion, mampu untuk berkompeten dalam bidangnya dan cepat memperoleh pekerjaan sebagai staf riset dan sains. Besar harapan saya, bioteknologi mampu mencetak banyak alumni yang mampu bersaing, baik di tingkat nasional maupun internasional.
Kami berharap bahwa lulusan program studi bioteknologi akan mendapatkan pekerjaan di berbagai bidang, khususnya di bidang bioteknologi dengan cepat, dan juga akan semakin sukses ke depannya. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Sometimes, a tiny stuff or things out of sight might turn to be main cause of every possible tragedy on Earth. Hence, a living creature should be observed for the sake of science.
Selamat datang di Fakultas Sains dan Matematika Universitas Diponegoro. Kami berkomitmen menjadi fakultas riset unggul berstandar internasional, mendukung Universitas Diponegoro bermartabat dan bermanfaat, serta mewujudkan World Class University ranking 500 dunia, dan mencetak lulusan berdaya saing global serta nilai moral dan etika yang kuat.
Accordingly, we put many scientists out, dedicated every single piece of our heart for each research. Assisted with sophisticated tools and advanced technology, no matter how complicated the situation is, it will be elaborated to bring out the solution. Compounds were studied, collected up to form a theory. Every single invention were integrated with another invention.
Melalui integrasi yang harmonis antara pendidikan, penelitian, pengabdian, dan kemahasiswaan, kami berkomitmen mencapai standar World Class University dengan keunggulan yang terukur dan berkelanjutan. Kami membangun ekosistem pembelajaran yang holistik, mengintegrasikan pengembangan akademik, keterampilan soft skill, dan hard skill. Penelitian dan pengabdian kami terpusat pada pengembangan solusi inovatif yang strategis dan berkontribusi nyata bagi masyarakat seperti mengentaskan kemiskinan, memperkuat ketahanan pangan, sekaligus mencetak generasi pemimpin yang visioner, berintegritas, dan berdaya saing global.
Giving wide possibilities for new circumstances to be explored, investigating the source and resources to find out novelty, and proceeding a sequence of practicums. Cultivating collaboration, domestically as well as abroad.
Ini adalah unit produksi, produk inovasi yang dihasilkan oleh Universitas Diponegoro dari Fakultas Sains dan Matematika yang berbasis teknologi plasma. Ada dua produk yang sudah dilisensikan oleh Universitas Diponegoro ke PT Dipo Teknologi, Zeta Green dan D-Ozone.
Considering the wide range of science, therefore, having strong mutual relationship with as many institutions as possible is the key for any kinds of successful research.
SEMEPSA, Research Center ini terbuka lebar bagi semua pihak ya, yang ingin mengembangkan riset-riset yang muaranya kepada pengembangan aplikasi biomonitoring, maupun juga pengembangan sustainable aquaculture. Kita sangat membuka lebar untuk kerjasama.
Anything impossible turn to be possible. Change something insignificant into something worthy. what was only a plan transformed to be a considerable execution. Upon every lecture, whether in the classroom or in laboratory, students were driven in an embedded discussion.
Selama perkuliahan di sini aku ngerasa dapat banyak banget ilmu, dapat banyak banget pembelajaran, mulai dari pembelajaran di kelas bersama dosen, dari sisi teorinya, tapi di sisi lain, dari sisi praktikalnya, aku juga dapat banyak banget ilmu melalui kegiatan praktikum yang menurut aku pribadi sangat baik sekali untuk menunjang aku di dunia kerja.
Ketika aku masuk ke Smart Building ini, pelayanan yang aku dapatkan sangat baik karena staf yang melayani sangat ramah dan siap membantu apa yang kita butuhkan. Di samping itu juga, fasilitasnya sangat memudahkan kita, seperti pintu yang dapat terbuka secara otomatis dan lift yang dilengkapi dengan sistem sensor. Terdapat juga ruangan yang cukup luas dan nyaman untuk mengikuti kuliah umum.
Supported with adequate facilities, the Faculty of Science and Math Diponegoro University adhere to inclusive education.
Di perpustakaan ini, aku mendapat berbagai macam fasilitas, yaitu ruang belajar yang nyaman. Selain itu juga, aku dapat dengan mudah mengakses media pembelajaran, seperti buku, jurnal, karya tulis ilmiah, yang dapat memperluas wawasan dan juga membantu aku dalam pengerjaan tugas aku.
Proved by providing enrollment for disabilities. Its students deserve the same opportunity to create their own bright future.
Dengan strategi peningkatan kualitas mahasiswa, optimalisasi fasilitas, dan pengembangan sumber daya manusia, kami menerapkan tata kelola efisien dan akuntabel, didukung teknologi informasi terintegrasi untuk memperkuat keunggulan institusi. Melalui kolaborasi internasional strategis dengan universitas terkemuka dunia, serta sektor industri, Fakultas Sains dan Matematika Universitas Diponegoro siap menjawab tantangan global, memberikan solusi berkelanjutan, dan berkontribusi pada masa depan Undip yang bermartabat dan bermanfaat.
We strongly believe that all the dream come true emerge from the soul that keeps vibrantly enthusiastic.
Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera bagi kita semua. Atas nama Sivitas Akademika dan seluruh warga Fakultas Sains dan Matematika, saya Profesor Dr. Widowati, Dekan Fakultas Sains dan Matematika Universitas Diponegoro, mengucapkan selamat atas diterimanya saudara-saudari di Fakultas Sains dan Matematika.
Assalamualaikum warahmatullahi wabarakatuh. Program studi Bioteknologi Universitas Diponegoro berdiri pada tahun 2018. Program studi Bioteknologi memiliki visi pada tahun 2025 menjadi pusat pendidikan, penelitian bidang bioteknologi sumber daya hayati yang berkontribusi dalam skala nasional dan internasional dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan. Untuk selanjutnya, misi dari program studi bioteknologi Universitas Diponegoro terdiri dari 1. Melaksanakan pendidikan tinggi jenjang sarjana bidang bioteknologi sumber daya hayati yang unggul, bermoral, beretika, berkompeten, berkarakter, dan berwawasan kebangsaan. Misi yang kedua adalah melaksanakan dan meningkatkan kualitas penelitian yang kreatif dan inovatif dalam pemanfaatan pengembangan sumber daya hayati yang berkelanjutan berdasarkan prinsip-prinsip bioteknologi. Misi yang ketiga adalah melaksanakan dan meningkatkan kualitas dan kuantitas pengabdian kepada masyarakat melalui pendidikan tentang pemanfaatan dan pengembangan sumber daya hayati yang berkelanjutan. Assalamualaikum warahmatullahi wabarakatuh. Saya Profesor Dr. Hermin Pancasakti Kusumaningrum SSI MSI selaku Ketua Program Studi Bioteknologi Universitas Diponegoro. Pada saat ini, Program Studi Bioteknologi Universitas Diponegoro berdiri sejak tahun 2018. Setelah 4 tahun, kami telah meluluskan sekitar 25% dari angkatan pertama. Dan dari lulusan program studi bioteknologi tersebut, ada beberapa mahasiswa lulusan kami yang telah memperoleh pekerjaan sebelum lulus. Kemudian yang membuat kami berbahagia adalah dari lulusan tersebut juga memperoleh penghasilan yang jauh di atas upah minimum rata-rata.
Hello everyone, my name is Nabila Maharani and I am a fresh graduate from Diponegoro University majoring biotechnology study program. I was pursuing this study for 3 years and 10 months and Alhamdulillah today I am graduating from Diponegoro University. When I was a student in here, I got a lot of experience such as laboratory experience, field sampling experience, and one cool experience that I got is that I have a chance to do a research collab with university overseas, such as Boston University. And with all of the experience that I got, I am optimistic that I could get a job opportunity in a research and development field. And do you know that this biotechnology study program has a really great laboratory facilities that you could do all of your research in here.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Muhammad Iskandar Zulkarnain, alumni dari program studi bioteknologi Universitas Diponegoro angkatan 2018. Pada program studi bioteknologi, saya telah mempelajari dan mengalami berbagai bidang riset, diantaranya adalah genetika, mikrobiologi, teknologi fermentasi, dan kultur jaringan. Saya yakin dengan pengalaman yang saya peroleh, saya dapat berkontribusi di industri hijau Indonesia. Bioteknologi keren!
Halo semuanya, perkenalkan saya Roma Redion, saya merupakan alumni program studi bioteknologi angkatan 2018 dan hari ini puji Tuhan saya telah diwisuda di tahun 2022 dengan predikat cum laude dan juga mendapatkan penghargaan dari dekan dengan meraih IPK tertinggi di atas 3,9. Selama saya berkuliah di bioteknologi, saya mendapatkan berbagai pembelajaran, terutama dari tenaga pengajar, dosen, bahkan dari kakak kelas kami juga. Dan ketika kita berbicara berkaitan dengan bioteknologi, di mana bioteknologi merupakan suatu ilmu yang mempelajari tentang pemanfaatan agen hayati untuk kepentingan barang dan jasa, saya sangat yakin bahwa bioteknologi ini merupakan ilmu yang sangat luas, ilmu yang mengglobal. Hal ini juga dibuktikan bahwa bahkan sebelum saya lulus dari bioteknologi itu sendiri, saya sudah mendapatkan pekerjaan, yaitu di Staff Research and Science di PT Haldin Pasifik Semesta. Berdasarkan hal tersebut, saya juga berharap bahwa bioteknologi juga bisa mencetak berbagai sarjana yang mampu kompetitif untuk mendapatkan pekerjaan yang terutama di bidang bioteknologi juga. Bioteknologi keren.
Saya Masda Manurung, ibunda dari wisudawan Romario Dion yang merupakan alumni dari program studi bioteknologi Universitas Diponegoro. Saya sebagai ibunda merasa bangga karena berkat berkuliah di bioteknologi, anak saya, Romario Dion, mampu untuk berkompeten dalam bidangnya dan cepat memperoleh pekerjaan sebagai staf riset dan sains. Besar harapan saya, bioteknologi mampu mencetak banyak alumni yang mampu bersaing, baik di tingkat nasional maupun internasional.
Kami berharap bahwa lulusan program studi bioteknologi akan mendapatkan pekerjaan di berbagai bidang, khususnya di bidang bioteknologi dengan cepat, dan juga akan semakin sukses ke depannya. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Hello, everyone, and welcome back. After this lunch break, I bet that you guys have been fully charged and ready for the next exciting session. Who's excited? Let me ask one of the participants. For the finalists, please turn on your camera and use the background that already provided. Okay, let me ask one of the participants. To finalist number 7, Sultan Rafi Abian Hidayat, are you there?
Yes, MC. Wait a minute, wait a minute. Let me prepare for
Hello,
my laptop,
Sultan.
please. Wait, wait. Okay, wait a minute. I'm still looking for my laptop here. Okay. Yeah. Yes, MC, I'm here.
I'm sorry, Sultan, but I cannot hear your voice. Are you already unmute your device?
Wait. Can you hear my voice? Wait, can you hear my voice clearly?
I'm sorry, Sultan, but I cannot hear your voice.
Okay, wait.
Maybe. Can you speak louder?
Hello, hello.
Okay, finally.
Okay.
Okay, finally I can hear your voice.
Okay.
So, Sultan, how do you feel after attending the webinar? Are you still excited for the next session?
Well, my patient is just like getting high, until right now. I'm still excited to hear all of the events until right now. Like the webinars are very insightful, I like how the professor giving the materials that share, and I'm interested about how she mentions about the omics in the oceans, because I'm currently studying in omics, and it's kind of giving me such a great insightful about the not just only the land species, but in ocean too, that we need to care full. And then for the professor of the some who give the materials from the University of Malaysia Sabah also give some great insightful too about the healthcare systems. All of the participants have given questions very clearly, and I like those the questions very well, very insightful too. And until right now, I'm still exciting for waiting the informations, the awarding and the winner announcements.
Wow, what an interesting answer. I hope the webinar earlier can help you in the future, since I heard that you also learn related things to the webinar, right?
Yeah, because I'm currently in a third year of my student year. I'm the third year here, undergraduate student, and I'm still like learning about the omics because there's like four terms like proteomics, metagenomics, genomics, and the others. And I'm quite a little bit interesting about how the professor mentions about the oceans omics, like she mentions about the eDNA too. That's a great innovation that has been currently very trending right now in biology materials and biology study here.
Whoa, okay. So that means that the webinar earlier was relevant or related to you, right?
Yep,
Okay,
exactly.
so I hope you keep excited until the end of the session. And thank you for your response.
Okay, thank you so much, MC.
Now it's time to move to the highlight of today's event, the announcement of the winner, which is what we've been waiting for. May this ceremony inspire and motivate us all. Okay, so we have two special awards to celebrate in other important categories of this competition. We will soon announce the recipients of the Best Presentation and Best Poster awards. And now it is my pleasure to announce the winner of the Best Poster award. This award recognizes the participants with the most compelling and informative poster. The Best Poster award goes to, drumroll, okay, there's actually a sound of drum roll, I guess. So, the winner, the Best Poster award goes to Tevika Prista Anindita from UPN Veteran East Java. Wow, congrats to Tevika. Wow. Oh, well, I can see that the poster is super good. Before that, I would like to invite Ms. Felsia to give the certificates to the winner. To the winner, please turn on your camera.
Okay,
Okay, congratulations for
semi team.
Okay,
For as the team leader, I congratulate your team because you already got the best presentation. Sorry, best poster as the winner.
Maybe for
Maybe for
Seta
Seta can open cam.
okay.
Okay.
Okay, then
Okay, then to the
as
winner,
we
as
go
we
into
go
the
into the
photo
competition,
session, prepared. Okay, we'll take some picture first. I'm going to count it down in 3, 2, 1, smile. Okay. Thank you, Stefika, and congratulations. Okay, once again, congratulations on Stefika on snagging the Best Poster Award. Next, we will announce the winner of the Best Presentation Award. This award recognizes the participants who delivered their ideas with clarity, confidence, and also impact. The Best Presentation Award goes to, drumroll, Alvin Hasrat Lose from the University of North Sumatra. Whoa, congratulations, Alvin. Alvin, you can open your cam and use the photo again. Okay. Before that, I would love to invite Miss Amel to give the certificates to the winners.
Yes, before I wanna congratulate you and your team for winning, and hopefully this can be an idea that is realized sustainably. And I also wanna say thank you for joining Aspire 2025.
Thank you.
Okay, thanks, Miss Amel, for the speech. To the winner, to the audience, please switch on the camera feature as we will have a photo session again. Okay, I'm going to count it down in 3, 2, 1, pose. Okay. Big congrats to Ovin on winning the Best Presentation Award. Now, we'd like to hear from our winners. To begin, we invite the best poster or best essay winner to deliver their speech. It's either Ovin or Stefika. Okay, so I still see that Stefika is still turning their camera on. So, Stefika, can you give us some speech, please?
Okay, maybe I can say thank you to the committee and to everyone in here.
Okay, thank you, Stefika, for the speech. And maybe it's now Ovin turns. You can give us some speech.
Thanks for the opportunity that given to us. I'm Alvin Hasalasa and my team, Agnes Kezia and Asti Amanda from Skowino Team Universitas Sumatera Utara. We want to say thanks to committee and also Universitas Diponegoro and Universitas Sabah Malaysia has made this competition. I hope every idea that we deliver, it will be had impact to students and also Indonesia and ASEAN. That's all. Thank you.
Thank you very much, Ovin. Okay, once again, congratulations to the best poster and best presentation winner. Okay, so let's move on to the next session that we've all been waiting for this exciting moment, and I'm sure you're also curious to know who the winners of Aspire 2025 are. So without any further ado, let's strike in. Okay, the third place winner goes to Ananda Muhammad Raka Putra from Diponegoro University. Congratulations, Ananda Muhammad Raka. Before that, I would love to invite Miss Adeline to give the certificates to the winner.
Okay, maybe I would congratulate to Ananda and the team. But Ananda, is there in the Zoom? Can you? Okay,
I'm
Alicia.
sorry, our team
Okay,
leader is
congratulations,
Veronica.
Alicia and team. We really appreciate your work, and we're looking forward for your team another paper. It is very insightful. Thank you.
Yes, thank you so much.
Okay, thank you to Miss Adeline. To the team who won, please switch on their camera and we will be having a photo session. Okay, I'm going to count it down in 3, 2, 1, smile. Okay, thank you. Okay, the next one is the second place. The second place goes to Derzu Torikali from Gajah Mada University. Congratulations, Derzu! Okay, before that, I would love to invite Miss Arina to give the certificates to the winner.
Okay, thank you, MC. Congratulations to Derzus team for winning the runner up position for this competition as one of your judges. I really appreciate your ideas and proposal is very great, and I hope that it will contribute to be a good solution for SDGs. Congratulations once again to their zoo teams.
Yes, thank you so much for the opportunity of this
Okay,
competition.
thank you, Miss Arina. And to the second winners, please turn on your camera since we're going to get a photo session. Okay. Okay, I'm going to take some photo. In 3, 2, 1, pose. Okay, thank you.
Congratulations.
Congratulations, Derzu. Okay, the last one, and I think this is the most waiting one since this is the first place. Okay, the first place goes to Ovin Hasrat Lasya from the University of North Sumatra. Congratulations, Ovin! Okay, before that, I would like to invite Miss Dita to give the certificates to the winner.
Okay, so Horace, Ovin, and team. So congratulations to the winner of the essay competition, especially Aspire. This is our first project, and so thank you so much to join our event, and congratulations again to Ovin and team. So you have a good idea for essay and a very insightful presentation yesterday, and all of the committee very enjoy your presentation here. So thank you, Ovin. Congrats.
Thank you very much.
Okay, congratulations, Ovin. Okay, so just like usual, Ovin, can you turn on your camera? Oh, okay. Let's take some photo session first. Okay, I'm going to count it down in 3, 2, 1. Smile. Okay, thank you so much, Ovin and Miss Dita. And now, let's give our winners a moment to share their thought with us. We can wait to hear from our winner speech. Maybe we can hear from the third winner first. Okay, for the third winner, the time is yours.
Okay, thank you. First of all, I would like to thank the judges and the organizer for appreciating our work, and also our mentors who guide us throughout this journey. Uh, Not only this competition taught us about science, but also about teamwork and persistence. It has taught us the importance of creativity and problem solving throughout SDG, encouraging us to think critically and apply innovative approach to achieve meaningful results. So thank you very much for this opportunity.
Okay, thank you, Alicia, for the speech. And maybe moving on to the second winner.
Yeah, I'm really grateful for this opportunity, and to be honest, I don't really expect to win any category or even get to runner up or second place. Once again, I'm really thank you for this competition and opportunity.
Okay, thank you, Derzu. And last one, the first winner. Ovin, the time is yours.
Okay, thank you very much. It's a surprise to us. For the first, let's we introduce. I'm Ovin, and my friend Astri can say hello. Astri Amanda. Hello. And Agnes.
Hi, I'm Agnes.
Agnes. And Astri.
Hello.
Okay, thank you. So we are from Universitas Sumatera Utara. So say thanks to organizer and judges that gave the opportunity to us to present our idea, namely Fetispite. And so thank you so much. That's all. Thank you.
Okay, thank you, Ovin, for the warm speech. And well, in chat, I can see that there is Professor Dr. Suhaini. Okay, for the jury, is there anything you would love to say about the winners?
Yeah. Uh, Congratulations to all the winners and also all the participants. Don't be down because of you didn't win this time, but you can come back next year maybe. So it's really an excellent experience for me, and then I can see all your ideas is very excellent, and I hope next time I told Miss Dita yesterday that I hope next event we will like open the final event to all the biotechnology students in UMS and as well as in Undip and also in other universities in Indonesia. So we will get more audience and also from our student side we can help them see how other biotechnology students think and how they develop their biotechnology knowledge, integrating it into the more application side. Okay, so congratulations to all the winners. I hope to see you all again next year maybe. Okay, thank you.
Thank you, Dr. Suhaini.
Thank you, Dr. Suhaini, for giving a warm
Thank you.
speech. Okay, before moving to the next session, let's give our winners some warm applause again. Maybe an online applause? Okay. Once again, congratulations to the all winners. Your achievements are inspiring from human curiosity and perseverance. May this tribute fuel your passion to continue increasing your creativity and hard work. Well, unfortunately, we are at the end of the event. The event will end with a documentation session to capture this memorable moment. So let's get ready for a group photo. I would like to hand it over to the staff in charge. Okay, so now Aspire Asian Sustainability Pioneers Competition 2025 has officially come to an end. However, the achievement we celebrate today, the hard work, creativity, and dedication of our winners will continue to inspire us all. To our esteemed guests and participants, thank you so much for your enthusiasm and support through this competition. A special congratulations to all the winners. Your outstanding accomplishment deserve the highest recognition and applause. May this awarding ceremony serve not only as a celebration of success, but also as motivation for everyone to keep striving for excellence and making a positive impact. Until we meet again at future events, I wish you all continued success and patience in your journey. Future events, I wish you all be happy and go on and keep going to being patient in your journey. Before I officially close the event, let's say the tagline for the last time. Do you guys still remember it? Yes or not? Okay. When I say Aspire 2025, you can say vision in action, insight for impact. Okay. I'm going to count it down in 3, 2, 1. Aspire 2025, Vision in Action, Insight for Impact. Thank you so much for the audience and participants that already joining the events today, and congratulations to all the winners. Thank you.
Dalam kepalamu ribuan memori, mana yang membuatku seperti hari ini. Jatuh pertamamu dari sepeda, menampakkan hantu di rumah tua.
Serpihan berbahagia atau justru buatmu merana. Jika ada ingatan yang terus menghangatkan dirimu, jaga apinya, hadapi hari, teruslah kau begitu. Jika ada pedih yang panjang mengikat tubuhmu, percayalah dunia tak selamanya harus begitu. Karya wisata liburan pertama, kado dari mama dan papa, karena berhasil naik kelas walau rata-rata pas-pasan. Nekat pulang lewat jam malam, dilarang jalan-jalan sebulan. Setahun bimbingan belajar, kalah ujian sekolah. Serpihan berbahagia atau justru buatmu merana. Oh, pertahankan atau melupakan pilihanmu, kawan, tidak mengapa. Oh, tak apa-apa. Jika ada ingatan yang terus menghangatkan dirimu, jaga apinya, hadapi hari, teruslah kau begitu. Jika ada pedih yang panjang mengikat tubuhmu, percayalah dunia tak selamanya harus begitu. Dalam kepalamu ribuan memori, mana yang membuatmu di hari ini. Jika ada ingatan yang terus menghangatkan dirimu, jaga apinya, hadapi hari, teruslah kau begitu. Jika ada pedih yang panjang mengikat tubuhmu, percayalah dunia tak selamanya harus begitu. Jika ada yang kau pelajari dari masa lalumu, jaga apinya, hadapi hari, teruslah kau begitu. a sky, 'cause you're a sky full of stars. I'm gonna give you my heart, 'cause you're a sky, 'cause you're a sky full of stars. 'Cause you light up the path. I don't care, go on and tear me apart. I don't care if you do. 'Cause in the sky, 'cause in the sky full of stars, I think I saw you.
We were undergrad, stayed out late, never made it to class. Out of restaurant in a taxi cab. You were sweating bullets on the way to my dad's, and oh, you said, baby, think we're moving too fast. I swear the magnolias flashed a smile. And that's when you caught me, hoping you'd stay a while. Baby, I'd give up anything to travel inside your mind. Baby, I'll fall in love again come every summertime. Daddy taught me to choose it wisely, but you don't have to try. 'Cause baby, I fall in love every summertime. 25 and we're missing church. How about everyone we're hating at work? Dinner with your sister and the joke's gonna hurt. Cry the way home and you're putting me first. Oh, yeah. You just always know what to say. We're strolling down the boulevard, dancing under streetlights. Oh, yeah. Every year we get older, and I'm still on your side. Baby, I'd give up anything to travel inside your mind. Baby, I'll fall in love again come every summertime. Daddy taught me to choose it wisely, but you don't have to try. 'Cause baby, I'll fall in love every summertime. Every day summertime. Every day summertime. Every day summertime with you. Every day summertime. Every day summertime. Every day summertime with you. Didn't you hear Amanda's moving back to Colorado? It's 2013 and the end of my life. Freshman year's about to plummet just a little harder, but it didn't, 'cause we kissed on that Halloween night. I bleached half my hair when I saw.
I had a dream so big and loud, I jumped so high